It looks like I'm good on the audio. Hello and welcome back to, sorry, we got an echo. 1 minute. Okay. All right. We're good. We're good. We're good. All right. Hello and welcome back to all of you who joined us for the course. For those of you who didn't join us for the past couple of days, I'm Sean Gibbons, a faculty member and microbiome researcher at the Institute for Systems Biology, or ISB. ISB is a nonprofit biomedical research institute located in Seattle, and we're an affiliate of the Providence Health System. We focus on some of the most pressing issues in human health, including the human microbiome, as you're hearing today, but also cancer, brain health, aging, and many chronic and infectious diseases. ISB also focuses on K through 12 STEM education and beyond, as you see, in particular by addressing inclusion and equity for students of color, women, and other historically underrepresented groups in science. You can learn more about ISB by visiting our website at isbscience.org. As a nonprofit research organization, we rely on the generous support of our donors. We provide these virtual events free of charge to folks. So for those of you who have the means to help support us, we greatly appreciate any support you can throw our way. And there's a link at the bottom of our registration site to donate to our efforts here. So before we begin, I'd like to express our sincere thanks to the sponsors of this event who really make it possible for us to provide it for free to all of our registrants. Applied Microbiology International, who not only are providing financial support, but are also providing free memberships to their society to all registrants this year for the ISB Microbiome series. Also, the Environmental Health and Microbiome Research Center, or the Embrace Center at the University of Washington, and Science in Seattle. So thanks to those sponsors for supporting us. Because of their support, we're able to provide this to a wide audience from across the world. I think if we count it all up over the past 5 years, it's about 6,000 registrants from across over 80 countries. All right. And for those of you tuning in today, if you'd like to share your experiences, I know social media is a bit on the wane these days, but we do have a hashtag if you want to talk about the symposium or the talks or the speakers. It's #ISB Micro 24. So please feel free to share. You can tag ISB on Twitter or X at ISBSCI or ISBSci. All right. So the last couple of days was our course where we kind of give people a crash course in basic data analysis skills and a little bit on our metabolic modeling tools that we've developed here at ISB. But today is the symposium where you're going to hear from researchers from around the world working at the cutting edge of the gut-brain axis, which is our theme for this year. So to give a little bit of background on what you're gonna see today, we've broken things up into 3 sessions. Each session has 2 30-minute talks and then a 30-minute Q&A mediated by our session chairs. So in session 1, the theme is on early life nutrition and the developing brain. This is chaired by Crystal Perez, and our speakers are Dr. Zelena Kohli-O'Rourke and Sid Venkatesh. Session 2 is focused on stress, anxiety, and depression. This is chaired by Jacob Cavan, and the speakers are Dr. Nathaniel Ritz and Hannah Holscher. Session 3 is neurodegeneration. So as you can see, we've sort of progressed from early life, midlife, to maybe later in life. This is chaired by Dr. Noah Rappaport, and our speakers this year are Doctors Priyanka Baloni and Guang Xu. So 30 minutes each, and then a 30-minute Q&A. If you have questions for our speakers throughout their talks, please enter those into the Q&A box at the bottom of your Zoom. So just click on Q&A and you can type your questions in there. But be sure to type the name of the speaker and then a colon and then your question so that we can link questions to speakers, because sometimes it can be unclear who you're asking the question to. So just be sure to do that. All of these presentations will be available after the fact on our ISB YouTube channel. Today's event will be live streamed, and so that live stream should be available pretty much instantaneously after the event is over. But later on, we will cut these videos up into segments for each speaker and for each session, so you can more easily browse through the talks. So these will all be available for you to watch at will later on. If you have questions for the organizers throughout the event, you can use our Slack channel to communicate with the TAs and the organizers of the series. But if you want to ask questions of the speakers, you really should enter those questions into the Zoom, because the Zoom is the only thing that our session chairs are able to see while they're up here. So your questions should be entered into the Zoom Q&A. Great. Okay. So that's really all I have to say. I'm excited to hear these talks today. Thanks so much to all of our speakers and our session chairs for their hard work getting this all put together. And we are going to start exactly on time. So that's 9:15 Pacific time. So Crystal Perez will be up here at that time. But in the meantime, we'll take a few minute break, grab a cup of coffee or whatever, and we'll see you soon.
Good morning, everyone, and thank you again for joining us for our symposium today. My name is Crystal, and I am an MD PhD student in the Gibbons Lab here at the ISB, and I will be chairing this first session that is centered on the intersection of the nutrition and the developing gut-brain axis. I am very excited to introduce you to our two fantastic speakers today, Dr. Elena Coley-O'Rourke, joining us from UCSF in San Francisco, California, and our very own Dr. Siddhavan Kantesh, joining us from upstairs in Seattle, Washington. We will have both talks back to back, as Sean has mentioned, followed by a moderated question and answer session after both talks. So please use the question and answer function on Zoom to ask or upvote your questions. And as mentioned earlier, please add the name of the speaker you wish to address before your questions. And without further ado, I will introduce our first speaker. Dr. Coley-O'Rourke is currently a postdoc at UCSF in the laboratory of Dr. Jinhua Piao, studying neuroimmune interactions in prenatal development. She completed her PhD in neuroscience at UCLA in the laboratory of Dr. Elaine Zhao, where she examined how the maternal microbiome modulates fetal neurodevelopment and long-term behavioral outcomes in a model of gestational protein malnutrition. Today, she will talk to us about malnutrition and the developmental gut-brain axis. And I'll pass it over to you, Elena.
Thank you so much for that introduction. I'm really excited to be here today with you all to talk about my graduate work in Elaine's lab at UCLA. So just a very brief introduction. We know that there are trillions of microorganisms that live in and on us. They populate our skin and our respiratory, GI, and urogenital tracts. And they really provide just an incredibly rich and diverse amount of physiological potential to us. So in Elaine's lab, we study the gut-brain axis. And this describes how, throughout our lifetimes, the microbiome, we focus on that in the gut, interacts both directly and indirectly with ourselves, the host, in order to influence brain function through a variety of different ways. So this can happen through vagal and enteric signaling to the nervous system. It can happen through direct or indirect sort of secondary signaling systems like immune and endocrine, or through sort of direct bacterially derived or induced molecules and metabolites. And these sort of interactive processes have been implicated in a really wide variety of neurodevelopmental, neurodegenerative, and psychiatric conditions. So we know that many developmental diseases or neurological diseases have prenatal origins and are also sensitive to gene and environmental interactions. And the microbiome presents a really interesting interface of gene and environmental interactions because it's shaped both by host genetics and environmental exposures. And this can happen both prenatally via the maternal microbiome as well as postnatally. The lab has previously published findings that show that during gestation, the maternal microbiome can sort of communicate directly to the developing fetal brain. In this case, perturbing the microbiome by reducing diversity or depleting the microbiome entirely leads to really appreciable changes both in sort of direct neurodevelopment and then in long-term behavioral impairments. So we see really direct communication from microbially modulated metabolites to the developing fetal brain. But it's not clear sort of the degree to which this happens in a more clinically relevant model. So to address this question, I chose to focus on a very clinically relevant model of environmental risk, which is malnutrition. I think we can all appreciate that malnutrition during critical periods remains extremely globally prevalent and really devastating. It accounts for almost half of deaths of children under 5 and is related to a lot of really persistent conditions, including neurological and metabolic. We know that malnutrition can act on microbial structure and function, leading to persistent gut immaturity and metabolic shifts. And interestingly, an individual's microbial composition can interact with both how they respond to malnutrition and how they respond to treatments. And there's been a lot of work previously in this field. And we also know that malnutrition during critical periods influences fetal neurodevelopment. So this is happening at the cellular, circuit, and behavioral level. And really critically, these effects can persist throughout the lifespan, even in the face of nutritional rehabilitation. So there's really a need to understand the relationship between these two effects and how it can be harnessed for potential therapies. So we think that this could be happening through a variety of different mechanisms, including direct metabolite-mediated signaling from the maternal gut to the fetal brain, as well as through other signaling systems such as immune and HPA axis. So my project during my PhD aimed to identify sort of how gestational malnutrition affects both maternal and fetal health, and then kind of pick apart what the role is of the maternal microbiome and whether this can be harnessed to sort of prevent some of the neurological impairments we see with malnutrition. So this first question has been heavily published on previously, but not as commonly in a sort of strict gestational model. So that's what I'll focus on today. So this is a brief overview of my techniques. I'll dig a little bit more into these later, but I used a mouse model to model protein restriction. And really importantly, the protein restriction diet I used, PR, it's 6% protein compared to a 20% protein control diet. And the difference is made up by carbohydrates and not by fat. So these are isocaloric and fat matched. And that's really important because we know that high-fat diet during gestation is also a neurodevelopmental disruptor. So the first experiments we ran were using this protein restriction diet during gestation, and then just looking at sort of the gross phenotypes. So we see a really dramatic weight loss phenotype in the dams. So they lose weight across gestation, and this isn't accounted for by a difference in how much they're eating. We see that they have smaller litters and that they have this really elevated corticosterone phenotype. So they're, just very physiologically stressed. And this goes hand in hand with the growth restriction phenotype in the fetuses, where the protein restricted fetuses are smaller than the controls. And FGR is very well documented in humans and is known to lead to a variety of lifelong deficits. Another thing we observed was a dramatic shift in the fetal brain transcriptome. So we did RNA sequencing on whole fetal brain. And we find that some pathways of interest in particular are affected. So we see upregulated genes related to nutrient transport. And we see decreases in genes related to things like T cell differentiation and nervous system development. So we see some kind of broad shifts in response to protein restriction. And then I've just pulled out some genes of interest here. We see some related to serotonin signaling. So this is serotonin transporter right here is significantly decreased. And then we see a variety of different genes related to immune signaling. So some ILs are bidirectionally affected, related to axon guidance, and other kind of neurobehavioral effects. So in addition to these prenatal phenotypes, we were interested in seeing the long-term behavioral impact of this gestational protein restriction. So really importantly, at birth, we switched all of the litters to control diet. We really wanted to isolate the gestational period as the time of insult. And we also implemented a cross-fostering paradigm to pull out the effects of direct gestational diet versus any kind of persistent effects on the maternal side. So even though they were switched to controlled diet, we figured, they may still have some behavioral or other physiological effects of having been on the protein restriction. And we wanted to be able to separate out those effects in the offspring. So the first phenotype we noticed postnatally was that there was a really distinctive sort of bifurcation in survival percentages. So protein-restricted offspring that were fostered to previously protein-restricted dams had only about a 20% survival rate, so very, low. And then interestingly, control pups raised by protein-restricted dams had about a 50% survival rate, whereas control or restricted pups raised by control dams had 100%. So this tells us that the protein-restricted dams have a really hard time supporting a litter, and that in particular, this is the most challenging when the pups were also born to a protein-restricted dam. So we let these pups grow up to adulthood, and then we ran a variety of behavioral assays. I'll just talk about 2 of those today. So the first we ran was open field test, which measures, we call it anxiety-like behavior. So it's sort of exploratory. And what we found was that protein-restricted pups raised by previously protein-restricted dams, this teal group here, had decreased time and decreased distance in the center zone, and we interpret that as an increased anxiety-like behavioral phenotype. And interestingly, this was sort of driven in particular by the female offspring. So they had kind of the strongest anxiety-like phenotype. In addition, we used Barnes maze to study learning and memory. And this is based on training to identify a specific hole around the edge of the table in which the mice are able to escape. So they want to get out of the center and into the escape area. And when we did this over a period of acquisition trials, we find that, again, this PR to PR group had impairments in learning. So it took them across these trials a longer time to escape into this zone. And then when we tested them later for recall, we find that they have a sort of perseveration where they instead, so if we remove the escape zone, they kind of stick right around where it had been rather than searching around for other possible escapes. But they didn't show any difference in the number of errors they made before finding the zone. So overall, we find that gestational protein restriction disrupts the maternal physiological state, induces fetal growth restriction, and alters the fetal brain transcriptome. And we find that gestational PR plus associated rearing reduces offspring survival and increases anxiety-like behavior and cognitive impairment. So our next question was, what role, is there a role of the maternal microbiome? And if so, what is it? So the first thing we observed in the protein-restricted dams was that across gestation, they had reductions in diversity. And this went hand in hand with sort of alterations in specific microbes. So here we've identified 31 microbes that were significantly different across gestation. And interestingly, they primarily belong to Clostridia class. And we know that Clostridia are involved in protein catabolism, so it makes sense that these are primarily the microbes that are affected. And in the early postnatal period, we see that these microbial alterations persist. So until about a week postpartum, even after switching to control diet at birth, we still see some microbial alterations on the maternal side. And interestingly, these correlated with other physiological changes in the early postpartum period. So again, that all of the mice are now on controlled diet, but they're still experiencing kind of persistent effects of protein restriction. And one of these is alterations in milk nutrients. This was done in collaboration with Lars Bode at UCSD. And we find that in particular, milk oligosaccharides are decreased in dams that had been on protein restriction diet, and this is regardless of the condition of the pups. And milk oligosaccharides have previously been related to microbiome, and they're also known to be really neurodevelopmentally supportive. We also found a persistent signature of immune dysregulation on the maternal side. And so we see alterations in splenic immune cells, and we also see alterations in serum cytokines. And really interestingly, both of these measures were significantly correlated to the microbial measures. So we see a sort of interaction between protein restriction-induced microbial changes and then these other kind of physiological phenotypes. So we were then interested in how manipulating the maternal microbiome might influence some of these PR phenotypes in the offspring. So we used a broad spectrum antibiotic treatment to deplete the maternal microbiome, both before and throughout gestation. And when we do this, we don't see a difference in maternal weight loss. We see a little bit of a blunting effect of the maternal cord phenotype, and we don't see any significant difference in fetal size. So most of the kind of gross physiological phenotypes are unaffected by this microbial depletion. Interestingly, we saw a sort of distinctive effect on fetal brain transcriptome. So when we use antibiotics to deplete the maternal microbiome, we have a huge number of genes that are dysregulated in the fetal brain. And interestingly, there's actually quite a small overlap in the genes that are affected both by protein restriction and by antibiotic depletion. Again, just calling out some of these genes of interest. So again, we have some serotonergic signaling related genes, a few genes related to sort of different neurogenesis processes, hippocampal synaptic pruning, anxiety-like behavior. And, oh, I forgot to mention this earlier, but we also compared some of these transcriptomic changes to the Safari database from the Simons Foundation Institute. And we see that there's overlap in the transcripts that are affected by protein restriction and that are implicated in autism spectrum disorders. And we found the same with antibiotic depletion. So there's some overlap between antibiotic-related genes and autism spectrum disorder-related genes. And interestingly, genes that are implicated in all three, there's only 5 of them, and they were all dysregulated in opposite directions by protein restriction and by antibiotic depletion. So it seems that primarily depletion is having kind of separate effects when compared to protein restriction, but it does seem that there is some kind of modifying effect here where some genes are affected by both. So as we did before, we were interested in the sort of long-term effects of the gestational microbial depletion, and we performed the same cross-fostering experiments. Here we didn't see much of a difference in survival between PR and ABX-PR groups. There were no significant differences in anxiety-like behavior, but we did see an exacerbation phenotype with learning and memory behaviors. So we saw previously that protein-restricted offspring already had significantly worse learning and memory compared to control. And now we see that when we deplete the maternal microbiome during gestation, there's sort of an even worse phenotype in learning and memory specifically. So here we see that gestational PR disrupts the maternal microbiome both during gestation and for about a week postpartum. We see that microbial depletion has primarily distinct, but also a few overlapping effects on fetal brain transcriptome compared with protein restriction, and that there's sort of a partial rescue of some of the effects of protein restriction, while other phenotypes like the cognitive behavior are exacerbated by microbial depletion. So this tells us that there could be some sort of bidirectional kind of effect of the microbiome in protein restriction phenotypes, but it also tells us that not all of the phenotypes we're seeing with protein restriction are necessarily microbiome dependent or microbiome modulated. So finally, we were interested in whether we could sort of target the maternal microbiome or use informed interventions to ameliorate some of these protein restriction phenotypes that we observe. So we ran untargeted metabolomics on both fetal brain and maternal serum. Here I'll just show fetal brain. And we find a really clear separation between fetuses from control or protein-restricted dams. And we can predict the condition with 100% accuracy. So there's a very kind of clear distinction here. And we primarily saw differences in amino acids down here and then in lipids. And interestingly, with amino acids, we see bidirectional regulation. So this tells us that there's some sort of compensatory mechanisms or brain sparing going on here because we have a kind of blunt amino acid restriction in the diet, and yet we're seeing kind of bidirectional changes in the fetal brain. So that's just kind of an interesting aside. When we compare these metabolomic results to the antibiotic depleted fetuses, we see that there's a subset of metabolites that are being regulated both by diet and by microbiome. And these are the ones that we're most interested. So out of this common pool, we focused on metabolites that were significant across both tissue compartments, so across fetal brain and maternal serum, and that were reduced by protein restriction. And out of that pool, we identified 10 metabolites. So they were reduced by protein restriction. They were similarly regulated across maternal serum and fetal brain, and they were dependent on both diet and microbiome. And just a little bit of information about these metabolites. So 5 of them are branched chain amino acid metabolites. 3 of them are very well-known microbially sort of modulated or completely microbially created. And then 2 of them are a little bit less well understood. So we took this pool of 10 metabolites and we supplemented them to the protein-restricted dams throughout gestation. And when we did this, we saw an effect approaching significance where the supplemented dams lost less weight throughout gestation. We found a partial rescue of the maternal stress phenotype. So maternal corticosterone was significantly reduced by the 10M treatment, although it didn't reach the control levels. But we didn't see any effect on fetal weight. So we still see the same fetal growth restriction phenotype as before. And then once again, we used our postnatal and cross-fostering paradigm to look at long-term effects of this supplementation. Really interestingly, we see this huge support of offspring survival postnatally. So the vehicle receiving protein restricted group had that same 25% survival, whereas offspring that had been prenatally supplemented with a metabolite mixture had about a 75% survival rate. So something about the metabolite cocktail is very supportive of survival. We also see some behavioral amelioration. So recall that the females in particular had that anxiety-like phenotype following protein restriction. And here we see that with the 10M supplementation, that anxiety-like behavior is restored, specifically in the female offspring. And when we look at the learning and memory in the sort of testing phase, we see that in particular, males are having fewer errors. They're making fewer errors as they find their way to the target zone. So we're seeing these really kind of sexually dimorphic behavioral amelioration in response to the 10M treatment. So overall, we see that 10M supplementation rescues maternal stress, offspring survival, and anxiety-like behavior and recall without restoring fetal weight. And this is a really important point, I think, because increasingly, I think we're seeing in the literature that there's some sort of separation between weight restoration and amelioration of some of these other phenotypes that we see in response to malnutrition. So to recap, we see that gestational protein restriction induces changes in both the dam and fetus, which persist into adulthood in the form of behavioral abnormalities. We see shifts in the maternal microbiome, both during gestation and in the peripartum period, which go hand in hand with metabolomic changes. We know that the maternal microbiome is playing a role because depleting it exacerbates some of the protein restriction phenotypes and ameliorates others. And we know that this role is at least partially metabolite mediated because a group of metabolites ameliorate some phenotypes without affecting others. So I'll go through this section quite quickly, but just very briefly, we were interested in seeing whether some of these conclusions could be applied to a more translational kind of cohort. We were very fortunate to collaborate with Doctors Allison Chu and Kara Calkins at UCLA, who work in neonatology. And we were able to study some of their infants who have fetal growth restriction. And when we look at these infants in the early postnatal period, we find that the fetal growth restricted infants have a sort of immaturity phenotype in their microbiome diversity. So particularly at week 3, they have reduced diversity, which then sort of catches up to control levels. And at week 2 specifically, they also had significant increases in Staphylococcus taxa. And these changes weren't explained by other kind of sort of clinical variables that we looked at. In addition, we found that these fetal growth restricted infants had particularly reduced cognitive development using the Bayley scores across the first 2 years of life. And when we correlated these microbial measures back to these measures of neurodevelopment, we find some significant correlations between the two. So this is a very kind of limited study. So the sample size is small, the samples are quite heterogeneous, and we don't know the underlying causes of fetal growth restriction in these cases. We don't know about sort of their nutritional status. So it isn't a perfect match to the mouse study, but I think it's intriguing that even with these caveats, we're still seeing kind of similar patterns emerging with relationships between growth restriction, microbial shifts, and then cognitive development. So finally, I think that there are many questions raised by this study. I'm just going to very quickly go through. I think I have 1 or 2 minutes left. So one question is about kind of the fact that not all of the phenotypes we see seem to be metabolite or even microbially mediated. So I think it'll be really important to very clearly delineate which phenotypes are microbially dependent or modulated and which aren't. There's also, I think, many questions still about what exactly is underlying the behavioral phenotypes we see and how exactly the 10M metabolites are kind of restoring those behaviors. So we see a variety of different pathways that are implicated from our RNA-seq data. We also see that there's sort of differences in stress and immune signaling on the maternal side, which we know can influence fetal neurodevelopment. I think we have multiple indications for serotonergic related signaling, so that's something that it would be really interesting to follow up on, I think. Another really important question here is about critical periods. So we only had the diet and the intervention during the gestational period, but for translational validity, it's of course important to kind of extend the critical periods as much as possible. So I think more work, for example, testing the metabolites during the early postnatal period would be really critical. And then finally, we see a lot of sexual dimorphism, especially in the behavioral results. And it's not clear exactly what's underlying that, but I think that's another really important question to get at, exactly how we're getting these sex-based differences in behavior and an amelioration by the metabolites. So finally, I want to thank the organizers for having me, as well as thank my graduate lab, my collaborators, my funding, and acknowledge my current lab at UCSF as well. Thank you so much.
Wow, thank you so much for such a fantastic talk. I'm like looking for my little emoji reactants, but I'll just clap in real life. So we have some questions open in the chat. We're going to reserve these for the later portion during the Q&A session. But if you're still, marinating and thinking about the talk, please feel free to add some more questions. And then I will go ahead and introduce our second speaker, Dr. Sid Venkatesh, who is an assistant professor here at the ISB. His lab aims to define the general principles of molecular recognition in the gut and to elucidate the mechanisms by which microbial metabolites impact human physiology and health. For his postdoctoral research in Dr. Jeffrey Gordon's lab at Washington University in St. Louis, Sid used germ-free animal models for the development of a new class of nutritional interventions that target specific underrepresented members of the gut microbiota of undernourished children. He will talk with us today about microbiome-targeted dietary regimens to combat pediatric undernutrition. And all right, Sid, I'll hand it over to you.
Thank you. I hope you guys can hear me just fine. Thanks, Crystal, for thinking of this work, and thanks, Sean, for organizing this wonderful symposium. I also really enjoyed Elena's talk. I think it was a very fascinating topic and obviously has huge health implications. So I joined ISB just a touch more than a year ago, and I found that it's a wonderful place to do blue skies research, as well as research with translational implications. But today I'll be speaking about the first half of my postdoctoral career in Dr. Jeff Gordon's lab. And I'll especially talk about work that I co-authored with Jeanette Gehrig and Hao-Wei Chang, who were two exceptional PhD students. And I just want to begin by acknowledging their outsized contributions to this work, as well as Jeff and members of his lab. So just a roadmap of today's seminar. I'll begin by talking about the link between undernutrition and the gut microbiota. We've already had a marvelous introduction by Elena on this topic. I'll then quickly skip over to some mouse experiments which I conducted using a panel of commonly consumed, locally available food ingredients shown over here, and how we assemble these into interesting combinations of diets, which then resulted in the promotion of specific bacterial strains. And then we validated these effects in both mice and piglets, as well as evaluated other metabolic and host effects of administering these particular diets, which we call microbiota-directed complementary foods. Then finally, we were empowered to partner with the Gates Foundation to conduct a clinical trial, which in our case showed treatment efficacy of one of the MDCF formulations compared to a control diet and other MDCF formulations. And so this work has now blossomed into an entire movement in which now 20,000 children are being administered these foods. And I should just mention that my contribution to this field sort of ended at this point, and I then veered off to do other mechanistic work, but it's continuing on in Jeff Gordon's lab. So just a quick word about the fact that childhood undernutrition is still a very serious public health challenge. So in the year 2022, about 148 million children globally under the age of 5 happen to be stunted. And by that, what we mean is that their heights were at least 2 standard deviations greater than that of the median of a cohort of healthy growing children. And perhaps even more worryingly, 45 million of these children under the age of 5 were wasted. By that, we mean that they were enormously underweight for their heights. Now, might not surprise this particular audience very much, but about 45% of childhood mortality can be ascribed to undernutrition. And really, undernutrition is only one side of the problem. It turns out that with the availability of ultra-processed foods, which are very nutrient poor, you now have a complementary obesity pandemic, which is ongoing. And about 37 million children under the age of 5 happen to be overweight in 2022. Now, we've already heard Elena describe some of the positive effects of undernutrition. But one can ask the question, what does that have to do with the gut microbiota? So it turns out that undernutrition is not just due to food insecurity alone, but really is a vicious cycle that's driven by a number of environmental factors, such as immune dysfunction, enteropathy, poor sanitation, colonization by enteropathogens, and so on. And it turns out that over the last 2-3 decades, there's been a building of evidence, an accumulation of evidence that the gut microbiota play rather critical roles in the harvesting of key dietary nutrients. They set up rather formidable barriers to colonization by enteropathogens. They shore up barrier integrity. They sculpt immunity by activating certain immune cell subsets. And in this picture, we see how a number of metabolites, such as secondary metabolites, secondary bile acid metabolites, short-chain fatty acids, gut hormones, such as GLP-1 and PYY, are just some of the very myriad ways in which the gut microbiota impact human health, and particularly infant development. So given that the microbiota seems to be at the nexus of so many factors that contribute to undernutrition, central question that drives the rest of this talk is, can we somehow manipulate the microbiota in a very targeted way to improve health outcomes? But really, to begin this process, one has to define a baseline, which in this case is to try and understand the developmental biology of the gut microbiota. So to address this question, my co-authors and I looked at 5 cohorts of healthy children. Fecal samples were collected from birth to 2 years of age every month. Then we used V416S sequencing to try and determine if there's a shared program of development, perhaps a signature of microbial strains that characterize how a healthy gut microbiota assembles, and perhaps that results in its healthy functioning. And what we found that this was indeed the case. So in this particular heat map, you can see that each row in this case happens to be a bacterial strain, and each column is a month of fecal sampling. And what is clear from the heat map is that a number of strains, which are shown at the bottom over here, predominate during early life, whereas others seem to colonize the infant gut upon weaning from breastfeeding. So they sort of predominate during later years of life, and this corresponds to an increase in diversity and function of the microbiota. And what we and individuals before us in the Gordon lab showed was that, in fact, there seems to be a persistent microbiota immaturity in that children with severe acute malnutrition, or SAM, seem to have a lower microbiota age for being the same chronologic age. And I'd especially like to thank Satish Subramaniam for his contributions in developing this metric, as well as Jeanette. So then the question arises, fine, there seems to be a program of development in healthy children, but what happens with children in severe acute malnutrition? So we went ahead and observed 54 children who had been administered a ready-to-use therapeutic food. And what we found was that administration of this particular RUTF really did nothing to move the needle whatsoever, in that these children remained very severely stunted, exhibited persistent metabolic abnormalities. Elena has already mentioned the importance of branch chain amino acids, and we basically saw that they were significantly reduced in undernourished children compared to healthy children. Very, very importantly, a key mediator of growth signaling, the insulin-like growth factor, the levels of this particular hormone were significantly reduced in undernourished children compared to healthy children. So then, given the fact that we recognize that current ready-to-use therapeutic foods had never been designed to target the microbiota, we were inspired to develop microbiota-directed foods that would impact the developmental biology of the gut microbiota. So then we began to develop a pipeline, and that involved first isolating anaerobic strains from healthy and malnourished children, and then learning how to colonize germ-free animals with these strains, and then giving them diets that were combinations of these very delectable food items, which you can imagine was a rather appetizing prospect for all of us. So let's take the first step here, which was we managed to isolate 14 bacterial strains that depicted various phases of gut microbiota development. Similar to the earlier heat map that I showed, some of these strains predominate during early life, whereas some of the others that are, in fact, the targets of our work seem to sort of colonize these individuals upon weaning from breastfeeding. And it turns out that these particular strains are very severely underrepresented in the gut microbiota of malnourished children compared to healthy children, and thus represent our therapeutic targets. So once we isolated these strains, which was onerous, we then went ahead and developed very robust colonization protocols, in that we had to learn how to colonize the guts of germ-free mice with these human-adapted strains, which was really very challenging and honestly was the fulcrum upon which this work then developed forward. So then the next order of business was to identify commonly consumed, locally available food ingredients. And I then designed these random complementary food combination diets, or CFCs, in which each one of these 14 diets were randomly chosen or sampled food ingredients from these 12 ingredients. And each one of these 14 ingredients had 6 randomly sampled ingredients. So CFC1 was had sweet pumpkin, spinach, rice, and so on. And the size of the bubble over here indicates the weight fraction of that ingredient in that particular diet. So CFC1 is rich in spinach, CFC2 is rich in potato, 3 in peanut, and so on. So we then colonized 3 groups of animals with these particular CFC diets in a very, very randomized fashion. And so the idea was to try and minimize collinearity in the provision of food ingredients to these strains. And at the end of each weekly treatment, we collected fecal samples. And we went ahead and performed shotgun sequencing of these fecal samples. In parallel, we went ahead and also obtained the whole genome sequences of these anaerobic strains that we had isolated. And we could then map the next-gen sequencing reads to the genomes of these organisms to obtain a very, very accurate depiction of their representation in the community. So in this way, we were able to correlate the levels of the food ingredients using this sort of a matrix, along with the abundances of these strains during that feeding period. And that then led to this sort of a heat map, which basically showed that bananas, chickpeas, tilapia, and peanuts had very positive effects on many of those strains of interest that I had spoken about earlier. And moreover, if one looked at a rice gruel that is consumed by malnourished children in Bangladesh, which is where these field studies were conducted, we found that many of the components of that particular gruel had inhibitory effects on these strains. And so this then gave us the confidence to go ahead and try and validate some of these findings. And here you have a sort of a representative example of the awesome power of using gnotobiotic animals. So there are two groups of animals that are given a microbiota-directed complementary food, or an MDCF, in which we have assembled those 4 lead ingredients that I had spoken about earlier. We benchmark the performance of this MDCF with milk suji khichdi halwa, which is a rice and lentil-based therapeutic food, which is administered to children who have malnutrition in Bangladesh. And we had the colonized counterparts, so experimental groups of animals that were colonized with just these 14 strains. And so it's really important to note that these germ-free groups of animals serve as reference controls over here. So using a spike-in control in which we basically spiked in a bacterial strain which is known to not colonize the human gut. In fact, it's found in soil bacteria. We were able to get the absolute abundances of these strains upon feeding them an MDCF versus a khichdi halwa. And what we found was that the community biomass in MDCF-fed animals is almost double that of khichdi halwa-fed animals, shown over here. And moreover, many of the strains of interest, such as F. prosnitzi, which is an extraordinarily important member of the gut bacterial community, was significantly elevated in animals that were fed the MDCF diet compared to the khichdi halwa diet. So then what are the kinds of host effects that one observes when one administers a MDCF to these animals? Well, one can take a look at short-chain fatty acid levels, which are well known to be bacterial fermentation products. Our very own Sean Gibbons, who has sponsored this symposium, has done a lot of breakthrough work in understanding the consequences of these metabolites in humans. And what we find is that animals that are colonized with the 14 strains shown over here, and then fed the MDCF diet, have significantly elevated levels of these short-chain fatty acids, particularly in butyrate, which is well known to be an anti-inflammatory metabolite, as well as promotes the levels of specific immune cells of interest, such as regulatory T cells and so on. And moreover, we also saw similar effects when we developed targeted mass spec panels to measure the levels of branch chain amino acids and various catabolites of those aromatic amino acids. For example, this particular catabolite, indole 3 lactic acid, is very well known to engage the aryl hydrocarbon receptor on the surface of CD4 positive, CD8 alpha positive immune cells, which are known to be protected. Now, what are the other consequences of administering an MDCF? So when one does microcomputed tomography of femurs collected from these animals, one observes that animals that were fed the MDCF and that were colonized have a significantly elevated level in measurement in the cortical bone area, which is basically a measure of chondral growth. Perhaps even more critically, one finds that circulating levels of IGF-1, which was that key mediator of growth signaling that I spoke about earlier, that is in fact found at far more reduced levels in undernourished children compared to healthy children, is significantly elevated in these animals. When one then looks at various components of the AKT pathway, the AKT-mTOR pathway in livers, then using antibodies against phosphorylated components of this pathway, one finds that again, the MDCF diet in colonized animals has the highest levels of these activated forms of the mTOR component. And so this entire pathway basically follows a well-known paradigm that IGF-1 signals to result in mTOR signaling, which would have very important consequences for growth signaling. So then we asked, can we then recapitulate these effects in an animal model other than mice, given that there are many significant differences, as we all recognize, between mouse and man? And in this, we were very fortunate that one of my co-authors, Hao-Wei Chang, who is one of the co-first authors in this work, had already spent a lot of time and attention developing a notobiotic piglet model. And so we then designed a 4-component MDCF and compared its effects to a 2-component MDCF. So this particular MDCF has peanut flour, chickpeas, soybean flour, and bananas compared to this particular 2-component MDCF. And what we found is diversity matters, as in all walks of life. But then in this particular case, animals that are fed a 4-component MDCF have superior rates of weight gain and bone cortical volume. And many of our targeted strains of interest, in fact, are significantly elevated in the cecum of these particular animals. So then we were empowered after obtaining this proof of concept to then go ahead and conduct a clinical trial. We partnered with the ICDDRB, which is led by Tamir Ahmed, as well as the Gates Foundation, and designed 3 particular MDCF formulations. MDCF1 had all 4 lead ingredients, but also had powdered skim milk. MDCF2 had only the 4 lead ingredients, and MDCF3 had 2 of those lead ingredients. And so basically we tried to recapitulate the effects that we had seen in mouse and piglets. And what we found was, in fact, that the diet shown in red over here, which was MDCF2, is extremely adept at a number of things. It reconfigures the gut microbiome in these healthy children towards a age-appropriate healthy state. And that was demonstrated by Arjun Raman in a companion paper with us. And moreover, studies that were led by Robert Chen, who was a graduate student in the lab and a very close personal friend, showed that many mediators of bone development and neuronal development, in fact, were significantly elevated by the MDCF2 compared to the RUSF. And this was both in terms of levels of plasma levels of these mediators. And then at this point of time, I sort of veered off and started developing mechanistic aspects of this project. And in close collaboration with Jia Cheng, we found recently that, and this work is going to come out very soon, that in fact, one of the consequences of administering an MDCF diet, in fact, is that you are able to alter the levels of a gut satiety factor. And this seems to happen in a microbially dependent way. In fact, what we've shown is that there are novel gut microbial enzymes that regulate the luminal levels of some of these satiety factors. And as you can well imagine, this can have consequences for food intake in malnourished children. So stay tuned, because that work is going to come out in the next week. Following on, members of the Gordon Lab have gone ahead and shown that administering this particular winning diet, the MDCF2 diet, compared to a ready-to-use supplementary food, show or exhibit superior rates of weight gain in children with malnutrition. And what is really remarkable about that work is that, in fact, the caloric density of the MTCF2 diet is 15% lower than that of the RUSF diet. And yet, it has a superior rate of weight gain compared to children who are fed the RUSF. Work that has been led by a number of authors in the Gordon Lab over the last year have shown that these effects are accompanied by changes in a number of important bacterial strains that are associated with increased weight for length. And moreover, trying to understand the molecular gastronomy of this process, there seem to be important strains, Prevotella strains, that are crucial for metabolizing some of the carbohydrate components of these diets. And that is how these health effects are administered. So I just want to end in the last couple of slides by talking about the fact that next generation nutritional supplements should also try to take into account that although much attention has been given to the first 1,000 days of a child's life, in fact, there are other organs and organ systems that have been neglected. For instance, if you take the human brain, it turns out that 85% of the adult mass of the brain is attained within the first 3 years of life. But then there's a lot of remodeling, a lot of synaptic remodeling that takes place well into adolescence and young adulthood. In fact, synaptic pruning, which is important for the maturation of the human brain, seems to go well into later years of adolescence. And so perhaps one can think about using nutritional supplementation to try and augment the enormous nutritional and metabolic demands of the human brain. And so I just thought I'd mention that next generational nutritional supplements should probably look not just at the early life of a child, but also consider looking at young adults, because presumably, and this ties a lot with the work done in Elaine Shaw's lab, there seems to be a clear connection between nutrition and brain development. So then just a couple of concluding remarks. We've shown that a microbiota-directed complementary food is important in reconfiguring the microbiota and seems to have a number of very important host effects. I've commented on the fact that we have a paper that's coming out soon, which has important consequences for gut-brain signaling, in that it seems to be that there's a whole new family of gut microbial enzymes that regulate the levels of a particular gut satiety signal. And that particular signal is well known to be an endocannabinoid that signals to the hypothalamus via the vagus nerve. So, that could be one particular gut-brain consequence of MDCF administration. I've also spoken, sort of commented about the criticality of going beyond the first 1,000 days, just because of the fact that there are organs such as the human brain that have enormous nutritional and metabolic demands. And these demands go well into late adolescence and perhaps even early adulthood. And it could very well be that nutritional supplementation during those phases of life might have an impact on alleviating various psychiatric disorders and so on. And I'll just say that many of these concepts can actually be transferred to other health conditions. For example, we have ongoing work in which we are trying to transfer these concepts and understand how we can perhaps ameliorate many of the gastrointestinal and clinical manifestations that children in cystic fibrosis are known to exhibit. And so that's work that's ongoing in our lab. So at this point, I just want to say that it's been a pleasure to do this work in the Gordon Lab, and we're very happy to continue on various aspects of this going forward. I especially I've already spoken about the enormous contributions of these two individuals. Gia, shown over here, is my partner in crime on the work that's coming up. I'd like to mention that Robert Chen was very instrumental in some of the work that we did, as well as a number of other people in Jeff's lab. I'm enormously grateful to my very young lab. We are now working hard on a number of enzymology and cystic fibrosis related projects. And I'd especially like to thank my mentors, Jeff Gordon, Mike Barrett, Paul Binash, Charlie Rice, and Mark Byrne, who've really set an example in my life and shown that science really should be conducted for the benefit of humanity. I think this is something which, we live our lives by, and it's probably one wonderful way by which we can change the world. So at this point, I'd just like to thank you all for your attention. Thank you.
Thank you so much to both of our speakers for such fantastic talks and a great start to our morning at the symposium. The question and answer is still open, so feel free to pepper in questions as we go ahead and get started. But I will be alternating questions to both of our speakers, so we can go ahead and start off. Let's see. Let's start with a question to Dr. Coley. Is it known what are the mediating factors of the effects of the maternal microbiome on fetal brain development?
Yeah, so I think that this is an interesting question. I think right now there is a variety of different sort of pieces of evidence suggesting a bunch of different pathways. So some earlier work from Elaine's lab, led by Helen Vong, that I showed at the beginning of my presentation, found a really direct relationship between maternal microbially modulated metabolites. She did sort of ex vivo studies where she dosed in the metabolites directly to fetal brain explants and found an effect. So I think sometimes we see that there is really just a direct metabolite mediated signaling effect. But there's a variety of other, studies showing immune mediation, showing, epigenetic modulation. So I think there are so many different pathways by which the microbiome can be influencing fetal brains, some very direct, some kind of more secondary. So I think that, all of these sort of individual studies are really exciting, but eventually we'll need some really integrated work kind of getting at maybe the different contexts in which, one mechanism or the other would be underlying phenotypes, or kind of in some way, integrating all these different pathways that we know can play a role.
It's certainly very complicated. Awesome, great. So now for Sid from Sean, I'm guessing that there is some heterogeneity in child responses to the MDCF diet, despite the average population scale effect, or some kids might be non-responders. So if so, do you think alternative complementary foods might rescue these non-responders, or do you think that you'd need to add a probiotic intervention to resolve the non-response?
Right, and you're absolutely right. There's there's the top and bottom quartile of response. I think it's fair to say that, as the administration of these therapeutic foods take place, there'll be a lot of lessons learned in terms when we look at the metabolic effects in these children, and that would help us fine-tune the development of MDCFs. Regarding your thought about probiotic supplementation, if I heard that right, well, that's a little bit of a challenging point over here, because we're talking about a highly vulnerable population, which is malnourished children. And so we're going to have to be absolutely sure that any probiotics that we are putting into these children does not have any other ill effects. In fact, one of the major challenges seems to be that introducing strains contributes to antibiotic resistance. And so we'll just have to be extraordinarily careful about how we go about that. And the reason why we started with a nutritional rehabilitation, I believe, is just because of the prospect of it being safe and well tolerated, as well as the fact that you can be quite compliant when you're taking a food supplementation. But of course, your point is very well taken. I think eventually, if one cannot reset the microbiome back to a healthy level, you would need to introduce either probiotics or postbiotics, that is, metabolites, beneficial metabolites of the kind that Elena mentioned, that can be directly administered to these children.
Thank you. All right, I think I'm going to continue by looking at questions that are upvoted. So for this next one, Dr. Coley, are there differential postpartum behavioral alterations in the PR dams compared to control dams?
Yeah, this is a great question. So this is one of the reasons why we wanted to do cross fostering, because we were concerned that there would be differences in maternal care. I looked at a bunch of different maternal behaviors. We found a difference in nesting very early in the postpartum period, where PR dams sort of were worse at kind of building nests and cultivating their pups. What else? There was no difference in retrieval behaviors, and there was no difference in sort of time spent on and off nest. And the nesting behavior was restored. I saw it at P0 and P1, and then it was restored by P5. So it does seem like there might be a little bit of a maternal care phenotype very early on, but then that does seem to be restored a few days after switching to controlled diet.
Gotcha. Thank you very much. Great. So this next question for Dr. Venkatesh, why do you think the addition of milk powder in the MDCF1 had such an effect? Was it simply because it reduced the amount of other key ingredients?
Please call me Sid. Well, that's an interesting question. I think in this case, it's because the milk powder actually promotes the absolute abundances of some of the bifidobacterial strains, which are highly milk adapted. So there's a whole body of work showing that these strains that are associated with early life, such as bifidobacteria, thrive upon the administration of breast milk in very young infants. And I think that's probably what is happening, is that in some ways we are delaying the maturation of the microbiota by administering milk powder.
Thank you very much. So next question for Dr. Coley, if you could speculate, do you think it's feasible that the Western diet could be recapitulating some of the microbial metabolic signatures of protein malnutrition? Have you seen evidence for this in the literature compared to your studies?
Yeah, this is another really interesting question. So, there are studies showing that Western diet, which is usually high fat, high sucrose, does have neurodevelopmental and behavioral effects. Whether they're exactly the same as what we would see in protein restriction, I don't think what I've seen is sort of exactly, recapitulating, but I think if we think about, diets in terms of ratios, if you have high fat and high sucrose, you probably have lower protein anyway. So I think we have to kind of start thinking about differences in diet as sort of ratios of nutrients rather than, a real binary of high in something. So I think kind of, again, this is a matter of right now we have studies showing kind of distinct phenotypes in a variety of different contexts. And what will be really important going forward is sort of integrating these findings and thinking about, are there signatures that are common to a variety of different dietary perturbations, or are we mostly seeing microbial changes or other phenotypes that are really distinctive to specific types of diet perturbations? So as of right now, I think we know that Western diet does have effects in similar domains, but I don't think it's necessarily the case that we would see exactly the same types of phenotypes.
Thank you very much. And now a question, I suppose for both of you, if you'd like to answer it. So from Mike, am I right in thinking that either of you, when you mentioned the gut microbiome, are referring to only bacteria? If so, is there any evidence that archaea or eukarya, such as protists, fungi, etc., might also be involved in these observed effects?
Elena, do you want to take a stab at this?
To speak for myself, we only looked at bacteria, but I think that is a really interesting question. I've seen one paper that looked at bacteriophages in stunting, and found some differential effects. But I haven't looked at anything besides bacteria.
And I'll just add by saying that although we also did not look at the effects of those other kingdoms, it turns out that I think it's now pretty well established that there are a number of gut viruses, gut fungi, etc., which are able to provide a colonization resistance to other predators, so to speak. And so, I think it's definitely a field for tomorrow, where some of the computational as well as experimental tools that have basically been invented to study the gut microbiota will now be, in fact, transferred to some of those other kingdoms of life.
Yes, it's definitely a very complicated question. We're only looking at one little puzzle piece. Awesome. So for our next question, Dr. Coley, were there sex differences in the offspring's serum metabolome? Is it possible that the exacerbated or persistent behavioral effects in female offspring may be immune mediated?
Yeah, so the only offspring metabolomics I did was in fetal brains. So we didn't know, and we pooled multiple fetal brains together. So I don't know the sex of those fetuses. I think down the line, it will be really important to understand what is underlying that sexual dimorphism. I think the idea that their sexual dimorphism in response to environmental stressors is not at all new. I think that's very well sort of established. But the underlying mechanisms, I think, aren't super well understood. So it's possible that there is an immune factor here, because we did see immune dysregulation on the maternal side. So it's possible that that is sort of differentially affecting offspring based on sex. I think there's also, potential differences in sort of placental function based on sex, and kind of SRY or sex hormone mediated effects. So we weren't able to really dig into what underlies that in this study, but I think that's a really important future direction.
Thank you so much. And now for Sid, do MDCF diets also help to reestablish hunger signaling, and would they be useful for treating adult malnutrition in the case of eating disorders, for example?
Right. Yeah. As mentioned, we have some work coming out on this, which at the moment I'm not permitted to speak about it, but that's just because of embargo reasons. But the central message of that work is that there seem to be gut microbial enzymes whose activity is stimulated upon MDCF treatment, and which can then regulate the levels of satiety factors, which are well known to signal via the vagus nerve to the central brain. And so it could very well be that administration of these MDCFs helps us to at least begin to solve a very paradoxical and difficult problem, in that children with malnutrition actually are quite anorexic, and they have a profound loss of appetite. And so stimulating that appetite is a huge challenge. It's almost as if their food intake and reward systems in the brain seem to have turned off. And so, it could be that administering certain foods, which then affect, metabolites that are satiety signals would be one mechanism to try and begin solving this problem. I think you also mentioned about adults, and since this work was done in children, I can't really directly answer that, but I will say that this strain is associated with healthy growth, and so I see no reason why that would not be the case.
Well, we will be very excited to look at your future work once it comes out. And then our next question for Dr. Kohli from an anonymous attendee. I was surprised by the high shine and diversity results in human infants, both the healthy controls and those with FGR. Even the control infants had a lot of staphylococcus and low bifidobacterium. Were all of these infants delivered by C-section?
So, no, they weren't all delivered by C-section, but these are infants that were preterm. So it's a little bit of a different kind of starting point. So all of the infants, both with and without FGR, were preterm. We looked at mode of delivery, and we didn't see direct correlations between the microbial findings and mode of delivery. So I think it's more just the case that this cohort in general had some abnormalities and, kind of more health problems than we would expect to see in just a full-term healthy infant cohort.
Thank you. All right, so now for Dr. Venkatesh, do you have any idea why some of the diets promote some good microbes while others don't?
Well, the screen that I conducted in mice was primarily to look for those sort of selective effects. And there was no guarantee that these ingredients would not result in the promotion of enteropathogens, which, thank heavens, is not the case. But I'm not entirely sure how to answer your question other than to say that when one looks at the molecular structures of food ingredients that make up our diets, there's probably a lot of commonality in terms of at least what would appear to be very different diets, but at the molecular level, they probably are quite similar. And strains have evolved with different populations, populations around the world. They've sort of co-evolved with the human GI system. And so, it could just be that they are particularly adept at metabolizing some molecular component of cassava versus rice, for instance. And so, but that's a highly speculative sort of answer, only because I'm being asked the question.
Thank you. Great. So now Dr. Kohli, for antibiotic treatment groups, were the mice singly housed or co-housed while being treated with antibiotics?
So prior to mating, the mice were housed in groups, and all of the mice within a cage were treated. So they were treated with twice daily gavages and then maintained on antibiotic water. So all of the mice in the cage were treated. And then following mating, they were single housed and maintained on antibiotic water as well. I assume this is getting at sort of coprophagic behavior and recolonizing, so that wouldn't be a concern because we're treating all the mice in the cage.
Amazing, thank you. And for Dr. Venkatesh, similar to a question that Sean posed a lot earlier, do you think that these MDCFs would have to be optimized for children in different countries that may harbor different microbiomes?
That might very well be the case. It turns out that they are now being administered. The field study started in Bangladesh. But then, and I'm not involved in any of this work, but now the Gordon Lab is basically partnering with the WHO and UNESCO and so on to administer these these foods which are formulated from, these four lead ingredients to begin with, at least, in multiple regions around the world. So in that way, one will be able to investigate generalizability of these effects. And as I said, there is no one size that fits all, and so we should always be open to the idea that any MDCF would require optimization.
Awesome. Dr. Coley, some of the components, like in your 10 metabolite administration, like indoxil and imidazole, have been associated with toxicity to the kidneys and liver in adult humans. But here they appear protective in the PR mice. Do you think there's non-linearity to the health effects of these compounds, maybe these molecules that are too low to indicate insufficient protein levels, but then too much, and then there's toxicity?
Yeah, I think that's definitely possible. We tried really hard to administer biologically relevant levels, so we weren't just like putting in a ton of these metabolites. So we were only restoring them to the control amounts that we would see in the control mice. So I think that's one important note. I think it's possible that there's sort of a dose dependency, and at one point it switches to be harmful. I think it could also be the case that it's just dependent on the time. So it's possible that at a certain point in development, you may need these metabolites, and then later on in life, they could become more detrimental.
Thank you so much. For our next question, this could be for both of you regarding the Mediterranean diet. What microbiome or bacteria flora could be aiding the beneficial neutral state and aging in this diet? Is there a way to mimic that by changing the Western diet?
Sorry, can you repeat that one more time?
Yes, I'm sorry. So I think they're asking more specifically about the Mediterranean diet and any, features of the microbiome that could be aiding its benefits to us. And is there a way to mimic that by changing the Western diet?
I'll take the first shot at this. It's actually quite challenging. I think there are studies that are happening, that are ongoing around the world, where one is looking at the effects of Mediterranean diet in humans. And at the top of I just can't recall specific strains that are promoted by the Mediterranean diet. But one would imagine that given it's so rich in many of the whole grains and fruits and vegetables and so on, that diversity of glycans perhaps results in the promotion of short chain fatty acid producers and bile acid producers, for instance. So I can only hazard a guess, because I cannot at this very moment recall details, but an educated guess would suggest that perhaps those kinds of strains are promoted, which are well known to be anti-inflammatory and have very beneficial effects. And over to you, Elena, if you have something to add.
Yeah, I think it's complicated. It's complicated to think about, because we know that certain diets support specific strains, but then also they contain, specific molecules that can be, processed by different bacteria differently. So if we're, thinking about what kinds of bugs do we want to support, but also what kinds of ingredients do we want to give them in order to, create metabolites, so I think it's complicated. And I think there are multiple ways to try to kind of intervene. So, using probiotics to like mimic what you would get with a Western diet. You can use metabolites to try to mimic what you would get. So I think you can kind of approach it in multiple different ways, but there are so many different sort of levels to diet microbe interactions that it's tricky to think about.
Lots to disentangle for sure.
And can I just add one thought over here, Crystal, if that's okay, if we are not running out of time? I think the point that Elena made, and it's something which I've also spoken about in the past, is, in my own case, we developed co-authors and I developed a microbiota-directed food. But of course, we know that, for instance, eggs are very, very important in terms of growth and may have nothing to do with the microbiota. One of the points which Elena had mentioned in her talk was that many of the effects might be completely microbiota independent. So I just want to acknowledge, that is certainly something we should always think about, that a nutritional supplementation should really attack as many aspects of physiology, microbiota just being one aspect.
Yes, very wise. Awesome. Next question for Dr. Coley was pup mortality or survival ratio explained by cannibalism by PR dams?
Yeah, so cannibalism was part of it. We did observe cannibalism by the PR dams, but we also saw some pups that seemed to be born dead, and then some litters that were just neglected by the dams. So we saw sort of a variety of different reasons that led to pup death, and then we included all of those in the sort of survival metric.
Thank you. Dr. Venkatesh, are the MDCF diets still effective in rescuing growth if you mix in other ingredients like rice or lentils that you showed down-regulated certain important gut microbes?
I have done experiments before where I mixed chickpeas and bananas with rice and lentils, and did see in mice, this is all mice, we did see dominant negative effects of the chickpeas and bananas in terms of the metabolites. So it does seem like they do exert those sorts of effects. But I guess more studies have to be done to answer that. I think the way that we would try and get around that problem is by prescribing MDCFs as nutrient supplements, so that even if children are continuing to eat all the things that they usually do, especially vulnerable populations like malnourished children who have a lot of food insecurity, that perhaps then having this supplement given once or twice a day would be sufficient to confer good health outcomes.
Thank you. Alrighty. Dr. Coley, do you know if there are critical periods during gestation when protein undernutrition has more pronounced effects on brain development and behavior in offspring? And this is my question, so maybe I'll add a little more context. I guess I was wondering if you'd, maybe explored switching the diet like halfway, maybe from protein restricted to control. Would you speculate that there would be sort of a rescue in the adult offspring like phenotype? Or have you, looked into that at all or seen any literature?
Yeah, that's a really good question, and I think really important for sort of translational applications of this work. In my study, I didn't do any manipulation of sort of within the gestational period. There's one paper I know off the top of my head that looked at protein restriction, and they found that even just in the first 3 days following conception, I think this was also in mice and maybe in rats, just the 3 days following conception was enough to instill these lasting, both neurological and behavioral impairments. So that's not the best news, but I think definitely more work is needed to really isolate. And I think in particular for specific phenotypes, I'm sure that different behaviors or different sort of pieces of neurodevelopment will be more or less affected by specific critical windows. In Helen Vong's paper, which I talked about earlier from Elaine's lab, they were looking specifically at the lammacortical axon outgrowth and somatosensory behaviors, and they identified a 1 week period within gestation. This wasn't with malnutrition, but with maternal microbiome depletion. They identified a 1 week period during which this effect was kind of relegated to that window of development. So I definitely think that there are specific periods that would be associated with, kind of pieces of the phenotype that we see. But we haven't investigated that in the malnutrition context.
Great, thank you so much. And now we are at time, but just because I want to squeeze in one more question, this is also for both of you. I think it'll be nice to kind of end on this question looking at timeline. Do you think these nutrition microbiome health effects will vary over the lifespan? Are there interventions that are optimal for the young, be bad for the old, and vice versa?
Elena, do you wanna, or should I go first?
I'm sure the answer is yes. I really don't know much about other sort of periods within the lifespan. But my assumption would be that, it's possible that different kind of processes or would be kind of more beneficial to be supported at different points during the lifespan. Like we were talking about the metabolites earlier, whether it's possible that specific metabolites during one period versus another could be more or less beneficial or detrimental. But I personally haven't looked at any other time points.
And going back to the thought I had about, and it's not an original thought, I should actually credit Mark Reichel and Manu Goyal, with whom we actually wrote this up as a review. Really, honestly, they should get the credit for sort of pontificating that, there's really a lot that goes on in the human brain after the first 1,000 days. And, this idea that the brain is basically a very metabolically demanding organ. And well into adolescence, you have extensive synaptic pruning which takes place. So that sort of remodeling of the brain is actually very critical and perhaps important for the onset of a number of psychiatric disorders, perhaps schizophrenia and so on, although there's a strong genetic component over there. So I'd probably say that we are going to almost certainly see supplements coming out in the future that target various ages. And this could just be one of them. And that's all that I can say about it, other than thanks for being such a wonderful moderator, Crystal.
Thanks. This is very interesting to me personally. So I thank you guys for being here. And sorry to our participants. I know I'm a little over time, but thanks for indulging me on my curiosity. We got to almost all of the questions. I won't answer the last one because we're already over time. So with that, I'm going to conclude the Q&A session and give my gratitude again to both of our wonderful speakers for their time and their expertise. We're now going to be taking a short break, but be sure to return at 11 a.m. for our next session on stress, anxiety, and depression. Thank you so much.
Hi, everyone. Welcome back. I hope you had a good break. We're able to get some stretches in and get excited for our second session today on stress, anxiety, and depression. So if you would please join me in welcoming our next two speakers for this session, Dr. Nathaniel Ritz, who's joining us from here at the Institute for Systems Biology in Seattle, and Dr. Hannah Holscher, joining us from the University of Illinois, Urbana-Champaign. So like the first session, please continue to write your questions into the Zoom Q&A, and be sure to write in which speaker your question is for. So without further ado, I'd like to introduce Dr. Ritz as our first speaker. He earned his PhD in neuroscience from the University College Cork in Ireland under the advisement of Doctors John Cryan and Ted Dinan, where he investigated the roles of the microbiome and virome in social and stress-related disorders. He's the recipient of the APC Microbiome Ireland Scientific Excellence Award. And in 2024, he joined the lab of Dr. Sid Venkatesh here at the ISB as a postdoc fellow, where he's elucidating mechanisms underpinning microbe-host interactions in an effort to find novel insights for therapeutic development. Today, Dr. Ritz will be presenting his talk, Stress Gone Viral, Gut Bugs That Can Impact Behavior in Preclinical Models. So with that, I'll hand it off to you, Dr. Ritz.
Okay, great. Well, thank you very much, Jacob. And thanks for updating my bio as well. I actually just won the APC award today, so much appreciated. Let's see, I assume you all can hear and see me, so I'll go ahead and share screen and get started. Okay, great. So yeah, today I'll be going over my talk entitled Stress Gone Viral, Gut Bugs That Can Impact Behavior in Preclinical Models. And I'd just like to state that this is all work that was done at the APC Microbiome Institute in Cork, Ireland. And since then, I've actually moved on to start a postdoc at the Institute for Systems Biology under the supervision of Sid Venkatesh, who gave a talk at just the previous symposium. So thank you again to both Sean and Jacob for inviting me to this. And yeah, let's get started. So to start off, a lot of the work that I'll discuss today all kind of takes place in the context of the microbiota gut-brain axis, which describes the communication between microbes in the gut and the brain. And I kind of wanted to point out that the microbial ecosystem in the gut can drive mood and behavior. And sometimes it kind of feels like science fiction talking about this subject. So I just want to point out, we're not talking about microbes specifically controlling your thoughts or leading to specific kind of ways to go about your life, but rather influencing how you feel, what your mood is, and in some cases, what kind of behaviors you may partake in. And it's also important to recognize that it can also impact other physiological systems. So we're kind of talking about the gut and the brain, but there are many different aspects of life, disease, and disorder that are affected by the microbiota. Now, a lot of the work that we know from the microbiota field and everything that's come out has actually come from a series of experiments that have been done using the germ-free mice, which are mice that are housed in captivity, and they are completely devoid of any microbes in the gut. And some of what we've learned for how this impacts the brain is we see increased myelination and myelinoplasticity of neurons. We also see increased neurotransmitter activity and related genes. We see reduced microglia maturity, which are kind of the resident immune cells of the brain. We also see increased neurogenesis, specifically in the hippocampus, as well as volume in the hippocampus and amygdala. We also see increased dendritic length and spine density, as well as increased blood-brain barrier permeability, which can lead to more metabolites and things being able to cross from the bloodstream into the brain. But rather than getting too far into any of those, I'd just kind of like to show this GIF of what a control versus germ-free neuron looks like. And it's very clear that you can see that there's an increased length of the neuronal branching, as well as, or sorry, there's increased length of the neurons themselves with increased spine density. However, there's decreased branching and connectivity and synapses to other neurons, which can lead to some of the different behaviors that we see. Additionally, this is a cross-section of a neuron, and you can see the drastic change between the myelin thickness, that's this dark circle going around the cell, and in the germ-free, it's much, much thicker, which is kind of an interesting finding in itself. So basically, when you see the germ-free model, you can tell very clearly that there's a lot of things that depend on the microbiome for normal development. Now, the next question that we're still working on in a lot of different ways is how is this communication system going back and forth? What is the crosstalk between the brain and the gut and the microbiota? Now, it primarily can be described in a few different ways. Microbes can actually have a direct impact on producing and converting neurotransmitters and neuroactive metabolites. They also produce different metabolites and products that can impact a variety of different physiological systems. They also can change the enteroendocrine system, which is the balance of hormones going throughout the body. Two classic examples are the microbiota having an impact on cortisol in humans or corticosterone in rodents, which is kind of the classic stress hormone. And lastly, the microbes can impact the microbiota gut-brain axis by modulating the immune system, which there's a lot of work actually coming out now and seeing how this immune regulation and development can cause all kinds of different changes. So specifically talking about stress-related disorders, we see a lot of comorbidity across diseases and disorders that we associate with the mind and the brain, and also with the gut. So these are kind of clear indications that we can expect that how your gut ecosystem is developing and its current state can impact different aspects of life. Now, specifically in stressed gut, we can see that it can lead to alterations in behavior, cognition, emotion, and nociception, or the sensing of pain. We also see altered immune activation and inflammatory regulation, as well as inflammatory and pathobiont-associated species generally being increased in stressed conditions. But how do we measure all these different parameters that are important for conducting this field of research? Here, I have a couple brief examples. First off, in A, this is the kind of classic microbiome studies where we look at differences in the fecal microbiota and associated metabolomics across two different patient groups. And sometimes the best way to do this is by longitudinal studies, because when you just take a short snapshot, you're just seeing a cross-section of a current state. But when you follow the microbiome through time, you can get a lot of really good indications of how it can be specifically affecting disease and health. Secondly, and something that we've been doing previously in the Cryon lab, is by testing in preclinical models, because it allows us to much more thoroughly control different aspects of experimental conditions. And basically, a lot of classic studies use something called fecal transplantation or fecal microbiota transplantation, FMT for short, where you actually will transfer microbiota collected from a group of individuals, and you can transfer that to different rodent models and see if there are any causal underpinnings to the fecal microbiota driving different diseases and disorders. And lastly, a goal is to then design different treatments that can have a positive impact. Some of the most common are changing diets or using prebiotics, which are kind of like food for the microbes themselves, or using probiotics, which are microbes that are generally associated with health. And with that, I'll just give a short example of a study that we got published earlier this year, where we looked actually in patients with social anxiety disorder, and we first categorized their microbiomes. So first, this is a beta diversity plot. So in the blue, we have the healthy controls, and in the red, we have individuals with social anxiety disorder, and each dot is a fecal microbiota that was tested by shotgun sequencing. And from this, we actually we selected individual donors, each one with a small icon here in the middle, to use as donors to transfer to mice to see if any aspects of social anxiety disorder could be transferred. And interestingly, what we found is in this model of social fear conditioning, there was a clear increase in social fear behavior. So quickly put, in the blue, we still have the controls, and the red, we have mice that received the social anxiety disorder, FMT. And basically, we saw that when mice had received the social anxiety disorder microbiota, they then had an increased social fear phenotype, which then corresponds to less time spent in social interaction. So this clear reduction is basically showing that. But as one of the questions brought up earlier, bacteria, which is what we largely talk about when we talk about the microbiota, they are not the only members. There are also viruses, there's archaea, there's fungi and protists. Now, bacteria do make up the largest amount of both activity and biomass in the gut, but the other microbes are certainly there. And the viruses in particular are quite interesting because the most recent estimates are showing that they're at roughly the percentage of 1-to-1 with bacteria in the gut. So they're quite numerous compared to the other members still, however, much smaller, of course. And of the viruses, they're predominantly composed of bacteriophages, which are viruses that infect bacteria specifically. And what's interesting is that these bacteriophages, or just phages for short, can have a variety of different impacts on their host organism. So the most common that we think of is the lytic cycle, where a bacteriophage infects a host bacterium, and it can lead to the cell lysing and releasing many more phages into the environment that then look for other bacteria to then infect. But actually more common in the gut are temperate phages, which can be both lytic or lysogenic. And lysogenic basically just means that when the bacteriophages infect the host bacterium cell, they either can incorporate themselves straight into the bacterial genome of the host or as a plasmid state. And what's interesting then is rather than producing a ton of different phages and causing the host cell to lyse, they can reproduce with the host over time. So it's interesting because we actually see more bacteriophage enter this kind of lysogeny or temperate state, where they then divide with the host and basically piggyback the winner. So whatever microbe is proliferating the fastest, they're able to hang around, and they divide each time with the bacterial progeny. And this can have a large effect on both the bacterial population as well as other members in the gut. Because when bacteriophage actually infects their host, it can lead to also a transfer of horizontal transfer genes. So basically, different functional parameters of the bacteria can change depending on the phages that infect it. So I'm going to kind of go back to chronic stress here quickly before we go to the next one. But the study that I'm mostly going to talk about today was making use of this stress paradigm. So what we did is we used something called the chronic psychosocial stress model, which basically corresponds of a series of social defeats and overcrowding of mice over a series of about 3 weeks, where they'll either receive a social defeat, where there's kind of a bully mouse that for a brief amount of time kind of bullies the experimental mouse. And then there's overcrowding times where multiple experimental mice will all then be put into the same cage, and kind of a overcrowding scenario. And then we compared just control, which received neither of these stresses, versus a stress group that went through this. And we looked at their different behaviors as well as some immune markers. What we found was that there's a reduction in social interaction following this type of stress. There's an increase in immobility time during the forced swim test, which is mostly seen as a stress coping type behavior. And there's also increased corticosterone, which was the stress hormone that we talked about earlier. We also saw some differences in splenocytes that were stimulated either with CON-A or LPS. But in addition to all of this, we not only saw differences in the bacterium, but also the virome. So here we have two plots, first off of beta diversity and then of alpha diversity below. And we see differences of the clustering between the control group as well as the stress group in the bacterium. But also when we looked at the virome, we saw a difference there as well. We did not see any differences in terms of alpha diversity, but we did mark a few individual bacteriophages that were either increased or decreased by the stress. And something else to kind of keep in mind is while we know there are many bacteriophages, it actually is very poorly defined at present. We actually have done a pretty good job of being able to identify different bacteria in the gut, largely thanks to the 16S ribosome, which you're able to then categorize many different genera of bacteria. But we don't have that same type of conserved gene in the virome, so each one needs to be individually characterized. And because of that, somewhere between 70 to 90% of the bacteriophages are actually unknown in the gut. So here we see that these top ones all fall into the Cotovirucetes group, while most of them are still unassigned. And this then led us to the question, could a virome-based treatment protect against the negative effects of stress? So using a tool called a fecal virome transplant, we autochthonously, which means just from self, transferred fecal virome transplant from a donor before they had any stress back to them as they were undergoing stress. So this is the study design for that next experiment. It's very similar to the previous. However, we removed the overcrowding phase because we didn't want any instances of coprophagy potentially affecting the microbiomes of the different individuals receiving their individual fecal virome transplants. So again, we had a control group, which received no stress and just a buffer oral gavage. And then we had a control stress group, which received the same buffer gavage, but it went through a chronic social defeat stress paradigm, where every day they went through the same social defeat procedure. And then throughout the study, we had a fecal virome transplant stress group, which then received its fecal virome transplant a few days apart throughout the study. Then on the 11th day, we started measuring different behaviors, the social interaction test to see how they interact with other mice, the elevated plasmase, which is typically just a anxiety-like behavior test, and the forced swim test, which again tests stress coping behavior. And then after that, we took more microbiome samples as well as collected tissue and did different splenocyte stimulations for kind of an assay of immune activity. And lastly, we assess outcomes of this study modulated by stress and restored by FET using something called an orthogonal contrast. So we were specifically looking for this kind of relationship where the stress group is moving in a different direction of both the control and the FET stress group. All right, so here's the first set of data. And again, just to kind of help you navigate this, because we're going to see a lot of graphs that are kind of in the same type. We have the control group, which is this blue one, followed by a control stress group, and then the fecal virome transplant stress group. So both of these went through stress and received gavages, while the control group had no stress. And both the controls just had the buffer gavage, while the green group received the fecal virome transplant. So what we saw is that there was indeed a stress effect in interaction and avoidance behaviors. And we saw a restoration of the stress locomotor activity in terms of distance traveled. The FET also protected against anxiety-like behavior and the elevated plasmase caused by stress. And then we saw that there was stress coping behavior that was not different across groups in the forced swim test. However, we did see a trend for it being an increase in corticosterone following the test, and which was somewhat reduced by the fecal virome transplant. Then we also looked at different immune cell populations, and we found that there were alterations in circulating immune cells, specifically in monocytes and neutrophils, and as well as the interferon gamma-induced protein 10 and CD62 ligand monocytes. We also saw that there were restoration effects of inflammatory cytokines released during splenocyte culture stimulation. And again, we used CON-A, which is a T-cell stimulant. Next here, we're looking at kind of the different characterization techniques we used on both the fecal virome transplant as well as the bacterium and virome of the mice. And this first image here is showing a cyber gold epifluorescence microscopy image of one of the fecal virome transplants. So if you see each one of these little dots, that's actually the nucleic acid of a virus or a bacteriophage in the gut. And we were then able to use that in combination with sequencing techniques to estimate the viral load of each fecal virome transplant. And roughly using both techniques, we could see that the total viral abundance and the FET were similar, although it did look slightly different depending on whether we used the epifluorescent counts or the sequencing counts, which kind of shows that these two different techniques kind of are looking for different markers of the viral load. And it's good to see that they are close, but there's definitely room for optimization of both. And I would imagine that is more closely somewhere in between of both of these markers. Next, here in this bar chart, we see the different FET profile of the different classes and families found in the gut. So again, in this case, all the blue are unassigned taxa. All the black fall into the class Caudoviruses, and then all the other viruses fall into the small bars in between. And lastly, then when we looked at the bacterium alterations in the guts of mice, we saw that there were 20 differentially abundant species, 15 of which had a reduced abundance following stress. So just showing here, this first column is all of the control group. The second column is the control stress group. And the third column is all the FET stress group. So looking for that kind of orthogonal change, we see that there's a reduction in 15 of these bacterial species with a reduction in stress that were then increased by FET. And there were 5 that were actually increased by stress and then reduced by FET. And then here we have a Sankey plot, which basically is something that we can use to test host-prey compatibility between the differentially abundant bacterial phyla and FET viral inocula. So it kind of shows how the differences of what we measured in the bacterium matches up with what we saw in the virome and what they would be predicted to align with in each of the different phyla. Next up, this is something that we included in the supplementary material for the paper, but I still find it actually quite interesting. There are a series of computational techniques called the gut-brain and gut metabolic modules, which can be used to test the specific genes that can be related to different aspects of brain sequences. So basically, you can look at a microbiome sample and see what type of functions are within those bacteria that can potentially impact the brain or the metabolism. So in this first one, what we actually did is we tested the gut-brain modules directly against the fecal virome transplants for each individual. And we were able to see differences in dopamine synthesis, as well as quinolinic acid degradation and S-adenosynyl methionine synthesis, which were all quite interesting. And then we also looked at the gut metabolic modules, and we saw a variety of differences across the different FETs. So this is kind of a more experimental technique and something I think can point towards the future when we're looking at this type of virome microbiota gut-brain axis mechanism. So each one of these could potentially have an impact by being transferred to different bacterial hosts through horizontal gene transfer, and potentially changing what type of function the bacterium then has. Of course, this is a bit theoretical, and we weren't able to prove this exact mechanism in this paper, but hopefully that's going to be something that we can do in the near future. Next, we did RNA transcriptomic sequencing on both the hippocampus and amygdala. And on these first two plots, they are volcano plots. And what they do is they show a transcriptomic analysis of genes that were altered by stress and then restored by FVT. And what's interesting is genes on the right side of this 0 line are all positively regulated by the FVT, while on the left, they're negatively regulated. And what was particularly interesting by this is in the hippocampus, we saw that it was roughly equal positive and negative gene activation, whereas in the amygdala, there is a skewing of it all towards the positive side. So that's kind of showing that there are a lot of genes that are affected by stress that are then returned to normal by the fecal virome transplant. Then we used a targeted functional enrichment analysis, gene ontology terms, which showed alterations of expression primarily in the amygdala. So each purple bar is for the hippocampus and each green bar is for the amygdala. And we saw a variety of differences from fear response genes to immune system processes, neurotransmitters, neurotransmitter receptors, as well as genes associated with stress response, social behavior, and viral processes. Again, these were all done on RNA sequencing in the brain. So with that, to kind of summarize everything we've gone through, stress causes alterations in host physiology and in the gut virome, and autochtonously transferring the virome back to the host during stress can protect against key brain and behavioral sequelae. And lastly, fecal virome transplant offers a potential novel therapeutic method for stress-related disorders. But I want to point out that this was specifically in mice, and more research is needed to see if this can be a viable treatment clinically. This was actually all published earlier this year in February. Here's the title here, in Nature Microbiology. So I would absolutely like to thank all of my co-authors and collaborators, as well as the whole CRIAN lab that was able to make this possible. And absolutely, I'm happy to be here now, part of the Venkatesh lab at ISB, and I'm very much looking forward to unraveling more of the mechanisms that drive this host microbe interactions. And with that, thank you again to the symposium organizers, and I'd be happy to take any questions, I suppose, later in the talk. Thank you.
Thanks so much, Dr. Ritz, for that really interesting talk on the virome. I feel like this is perfect timing, given that we just had somebody ask about other microbes that are not bacterial. Okay, please continue to write in your questions for Dr. Ritz in the Zoom Q&A. And then now, please join me in welcoming our second speaker, Dr. Hannah Holscher. Dr. Holscher is an associate professor of nutrition at the University of Illinois, Champaign. Hi, Dr. Holscher.
Hello.
And at the University of Illinois, Urbana-Champaign, she is also a member of the Division of Nutritional Sciences, the Institute of Genomic Biology, the National Center for Supercomputing Applications, and the Personalized Nutrition Initiative. Her innovative use of machine learning to identify microbial biomarkers of food intake and health earned her the 2017 New Innovator in Food and Agricultural Research Award and recognition as a 2020 National Academy of Medicine Emerging Leader. She's also received the 2021 American Society for Nutrition's Mead Johnson Young Investigator Award for her work on nutrition and the human gut microbiome. Her lab explores the connections between nutrition, the gut microbiome, and health using clinical interventions and computational approaches. Today, she's going to be talking to us about dietary and microbial connections to mood and cognition. So with that, I'll pass it on off to you, Dr. Holscher.
Excellent. Is everything coming through okay?
Yes, I think we
All
can hear
right.
you great. Thanks.
Wonderful. So thank you so much for the kind invitation to be a part of this symposium. And I'm really excited to be able to share some of the work that my laboratory has been doing basically over the last decade here at the University of Illinois. So I do want to start with some brief disclosures. My employment at the University of Illinois, I've listed my grant and research support, and I've bolded Tate & Lyle and the United States Department of Agriculture because I'll be specifically sharing some research that was funded by those two groups today, and then noting some speaking and travel honoraria from food commodity boards and private industry. All right, so my research laboratory investigates this connection between diet, the gastrointestinal microbiome, and health. And the other thing that I want to make sure to note is that I am a registered dietitian. And so the lens in which I approach scientific problems is very applied and translational. So I really love the previous presentation about the deep dive mechanistic insights into the connections between the gut and the brain, because we need preclinical research, we need computational research. All of this research has to work in concert so that we can move the needle forward. Where I spend all of my time is in the clinical domain. So we will do different types of clinical interventions, feeding studies, where we're investigating specific nutrients or specific food components like a prebiotic, a probiotic, or a fiber. We'll look at specific foods like those shown on this plate. We've done complete feeding studies with avocados, for example. And so in order to answer our research questions about how does diet affect health, and how is that mediated or moderated through the composition of the microbiome? Because a lot of my time is spent focused on non-digestible food components, namely fibers, that human enzymes cannot break down, but certain microbes can. And specific to the areas of health that my group mainly focuses on, we do a lot of cardiometabolic health research on individuals that are at risk for disease, so individuals with overweight and obesity. And then through collaborations with colleagues in the Department of Health and Kinesiology here at the University of Illinois, we also are able to extend our work and to look at the gut-brain axis. So we'll look at brain health, we'll look at executive functioning, and also thinking about mood and emotion. And so I'll be highlighting some of the work we've done in that area today. All right, so I really want to make sure that one of the key takeaways from this presentation is just the complexity of diet and how it is difficult to do these types of studies and be able to translate the research. And so one of the reasons is the way that we assess diet. So we will frequently need to utilize subjective questionnaires like diet records or food frequency questionnaires. And if you're like me, sometimes I have a hard time, or actually most of the time, I have a hard time remembering what I had for breakfast yesterday. And so when we do utilize diet questionnaires where you're needing to recall what you've consumed over a prescribed period of time, that can be quite challenging. And it's going to have limitations. And so we do need to rely on larger studies and then thinking about doing intervention trials to be able to tease this out in a bit more detail. So some of the research I'll be talking about today is thinking about diet in different ways. So we can look at diet in lots of different ways. So some of that is diet patterns, where we can use scores to characterize how closely someone is adhering to the Mediterranean diet pattern, for example, or another diet type, which is called the MIND diet. So this dietary approach is a combination of the Mediterranean diet and the DASH diet. And we're thinking specifically about specific components that are linked to cognition and neurodegeneration. So a key, some food components that are highlighted in the MIND diet are going to be green leafy vegetables and berries. And so these have been connected with different brain health outcomes. We can also look at diet through diet quality scores. So the Healthy Eating Index will rank how closely you're adhering to the dietary guidelines for Americans. And there are other quality index like the Dietary Inflammatory Index. We can again also do investigations looking at specific nutrients. The ones that I'm most interested in are going to be fiber and different types of fats, because microbes break down fibers to produce metabolites like short chain fatty acids. And fats are super interesting because of the human body secretes bile acids to emulsify fats. But we're learning more and more about how microbes are actually able to modify and degrade those bile acids, and they can have subsequent signaling actions within the gut and systemically that can regulate a range of functions, including glycemic control is one example. And then there are other components in the diet that we may be interested in. So probiotics, those live microorganisms that can provide a health benefit. Prebiotics, those selectively fermented nutrient sources that stimulate certain microbes and result in a health benefit, and then growing interest in fermented food. So all of these different components shown on this slide here have been investigated for their connections with brain health and cognition and mood. So one of the areas, as we're thinking about all of this, and it was alluded to in the previous presentation, so set the stage, so I'll just briefly go through this. When I'm thinking about the microbiome, I'm specifically quantifying the gut microbiome through the use of human fecal samples. So we have to acknowledge the limitations of using human fecal samples in that regard. The other thing that we'll very frequently measure are microbiome metabolites, namely short chain fatty acids and bile acids. And so we think about the importance of the microbes and their byproducts as they're interacting with the diet and the host, because these aren't only going to have effects within the gut, but they can have systemic effects. And some of the different ways that they'll act is because of the effects on inflammation. They can affect neurotransmitter secretion. So we can think about GABA that can come from certain microbes like lactobacilli. And we can also acknowledge that these metabolites will affect the secretion of different hormones. Incretin hormones like GLP-1 that can affect appetite, as well as serotonin and other hormones. And so, of course, these are going to affect gut health. The microbiota composition and its metabolic functions and metabolites will directly affect gastrointestinal health, but it does have systemic effects. And that can, when we think about brain health and cognition, we have ways we can query anxiety, stress, cognition, and depression is some of the work that we've done. All right, so what I'm going to do for the remaining presentation is walk through some of the different research that my group has been involved in that ranges in the level of scientific rigor for being able to make, our strongest conclusions. So I'm going to show some cross-sectional research that we've done, which is going to be more exploratory, and we have to always acknowledge that it's going to be at the bottom of that evidence period, where cross-sectional work is going to have the highest risk of bias and because of that exploratory nature. Moving into our intervention studies that we've been doing over the past few years, where we have very well-controlled randomized crossover designs, where we have an intervention and a placebo, and our conclusions for this can be much more robust because of the stringent design that we're using in our interventions. So to start with some of the cross-sectional work, Andrew Taylor from my group helped lead some of this, where we were cross-sectionally looking at some of the participants that had participated in research in our laboratory, and we were querying how their diet, using food frequency questionnaires and assessing that using the Healthy Eating Index. And we also were able to have, we had the DASS questionnaire. So this tool queries depression, anxiety, and stress, and there's 42 questions, and it'll can be used in individuals that are without cognitive or any type of diseases, diagnosed depression or anxiety. So it allows us to assess that, and then again, we're using the fecal microbiota as our marker. So what we reported in this publication in Nutritional Neuroscience was that there were associations between the Healthy Eating Index components and mood. So there was a connection between total fruit. It was inversely related to the depression scale, meaning that individuals that consumed more fruit had a lower score on the depression scale. We also found in this cohort that the dairy component was inversely related with stress. So again, as individuals more closely adhered to the dietary guidelines and were consuming those dairy components, they scored lower on the stress scales. Separately, looking at the microbiota, in this study, we found that there were connections between the microbiota and mood. Bifidobacterium was negatively associated with anxiety in females, and lactobacillus was negatively associated with depression symptoms in males. So one of the other cross-sectional studies that we have done was led by Tori Holthaus in here at the University of Illinois. And she was again using a cross-sectional design where we had a larger cohort in this regard, where there were 207 individuals without mood disorders. And we were again utilizing food frequency questionnaires and calculating their adherence to dietary patterns. So we were interested in the MIND diet because remember, this has been shown in other studies to be selectively associated with cognition and helping to support cognition and reducing neurodegenerative diseases like Alzheimer's. We compared also to the Mediterranean diet pattern score, the DASH diet, which is the dietary approach to stop hypertension, and then also the healthy eating index score. One of the key findings that I want to highlight from this study is we found that individuals that had the greater adherence to the MIND diet pattern, it was selectively associated with faster information processing in these study participants. But this association was not observed with the other diet patterns. So it seems that there was some sort of selective connection between adherence to this MIND diet and the information processing scores in this study. So moving into some of the longitudinal and interventional studies that have been done in the field, what has been reported in, and we summarize this in an older review now, where we were looking at dietary and microbial component connections to depression, anxiety, and stress. And so at this time, looking at the totality of the literature when we published this, there were certainly connections between diet quality, the Mediterranean diet pattern, and a lower inflammatory potential. So some of those longitudinal studies had shown connections in that regard. Now, thinking about the intervention studies, in this paper, what we were able to highlight was that there were, again, connections about how diet independently affects mood. Some of the nutrients that were investigated were unsaturated fatty acids, certain probiotics, improving psychological or biological measures of depression, anxiety, or stress in a lot of different connections. And then also that fermented milk containing probiotics could reduce physical symptoms of stress and prevent an increase in salivary cortisol and alter the brain network activity. So there has been so much work in this domain over the last few years, and this area is just continuing to be investigated. And so this is a highlight. It's not all encompassing, but some of the research that has been done, not just within my lab, but within the totality of the scientific literature to better understand diet, brain health, and the microbiota. So these are studies that include all of these components within human intervention studies. So the Mediterranean diet has been shown to improve cognition and change the microbiota in elderly populations. Fiber-containing and fermented foods can decrease depression scores, increase self-help, and increase alpha diversity in microbiota in women with obesity. Prebiotics, thinking about those selectively stimulated ingredients that will nourish the microbiota to support a health benefit. 5 grams of short-chain FOS and 7 grams of GOS improved anxiety and depression and enriched bifidobacteria. With a different fiber, polydextrose, consumption modestly improved cognitive performance and enriched specific bacterial populations in healthy females. And then there were other microbes, so L-caseae reduced anxiety and also enriched lactobacilli and bifidobacterium in individuals with chronic fatigue syndrome, so that probiotic approach. Diets high in fiber, this one's going to be much more recent, 2023. Fiber in fermented foods, and this was done, Kristen Bearding was with John Cryon's group when they were doing this work. So this was a very nice study showing connections between fiber and fermented foods and perceived stress. And then one that I find really fascinating that just recently came out of Dr. Kristen Verbecki's laboratory, where they were reporting that colonic butyrate administration modulated fear memory, but not acute stress response in men. And so you can see that these intervention studies really run the gauntlet, again, for these interventions, from dietary patterns to specific foods or nutrients, or as well as these probiotics, prebiotics, and fermented foods. So there's lots of research in this area with the interventions, and it makes me very excited for the continued work in the field. So what I'm going to talk about now for the next 2 slides are 3 specific intervention studies that my group has done specific to diet microbiota and cognition or mood. So the first one was looking at the effects of probiotics on cognition. And this was done here at Illinois, and Corinne Carnevale was the lead scientist here. And this was a randomized controlled crossover design study where we enrolled 26 healthy adults. So meaning these individuals could not be diagnosed with any type of cognitive disease or gastrointestinal disease. They consumed in a randomized order a fermented dairy beverage that contained probiotics, or the control beverage for 4 weeks. Each of the conditions were separated by a 4-week washout to make sure we had adequate time for those interventions to revert back. And then they participated in the other condition. So just to highlight some of the main findings from this study, we did see that the fermented dairy beverage increased lactobacillus and some specific bacterial strains, namely Lactobacillus zeae, which was one of the microbes that was present in the fermented beverage. So what you're seeing here is a list of the microbes that were specific in the fermented dairy beverage that we were investigating. And the control group is on the left and the probiotic condition on the right. And you can see there are numerically greater numbers for several of these, which you would expect from the probiotic being consumed within the beverage. We could detect those in the stool sample, but not all of those were going to be statistically different. For the cognitive outcomes, the main outcome we had here was with relational memory. And so here what we're looking at is a function to assess hippocampal function. And so shown on the screen here are some of the images that our study participants would see during these cognitive tasks. And so they would go through an original display condition, and then they would be allowed time to acquire that image and memorize, try to memorize that. And then they are asked, there's a bit of a delay, and then they're asked to move those images during the test phase, which is self-paced. They're going to move each of those images and put them back in the position where they found them. And there are different ways we can measure the accuracy here. You can measure misplacement errors, where you can see in this middle box here, where you can see in the light gray where the image should have been, and you can measure the distance for where it was placed. And then you can also look at it in kind of a larger context, a more broad or crude measure with just object location binding. And so if you're getting close within that circle, kind of a bit more moving towards that bivariate of, are they incorrect or correct, if they can get close to within that circle. And what we found was with the misplacement, when we used the more sensitive measure of that distance, within the pre-test to the post-test condition, there was no difference within the control, but within the probiotic condition, those individuals were able to have less misplacement errors, and that was statistically significant. And you can see a numeric improvement with the location object binding, but that did not reach statistical significance. So, one of the other things that I do want to mention here, though, with this particular study, we did not find any statistical relationships or correlations, mediation or moderation with the microbial and the cognitive outcomes in this particular study. There was a change in both the microbiota and a cognitive measure, but those were not interrelated. One of the other studies that we've done here, we were specifically interested in the effects of prebiotics on cognition. And this was led by one of my doctoral students at the time, Anne-Marie Meisenheimer, who's now at Cincinnati Children's as a research scientist. And Anne-Marie was focused on prebiotics. So specifically, we were utilizing 5 grams of fructooligosaccharides and 5 grams of galactooligosaccharides mixed within a dairy beverage, so 1% lactate, for 4 weeks. And then the individuals would pass into the other condition after the 4-week washout. So for this particular study, we did see, as would be expected, this prebiotic effect with VOS and GOS. There was an enrichment in the prebiotic condition in blue with bifidobacteria at the family level, at the general level, and then several of the species were enriched with the prebiotic condition as compared to the control. So we did see the dose was adequate for that prebiotic effect. However, for the results in the cognitive domain, we found there were largely null findings of the prebiotics on the other markers of biological and behavior markers of stress. There were also no effects on inflammation, mood, or sleep. The only exploratory finding that was found from this one was when we were specifically looking at the responders and the non-responders for the bifidobacteria, meaning that for the individuals that had an increase in bifidobacteria within their microbiota in the prebiotic condition, there was a trend for having a more positive response to positive image valent, to positive images. So this picture shown here on the left would be an example of a positive image, a baby sitting with a bear. And the individuals, there was a correlation with the amount of bifidobacterium and the valence ratings that these individuals would have and report. And so again, with some of these largely null findings, one of the take-homes that I think is always really important to acknowledge is that it is difficult to impart a physiological benefit in individuals that are in the normal domain, right? They're not suffering from a cognitive disorder. They're not diagnosed with any types of diseases. And so moving the needle on these can be quite challenging. The other thing to point out, especially with these markers of stress, we measured 24-hour urinary cortisol in this study, as well as the one I just mentioned on probiotics. And again, with that 24-hour urinary cortisol, this really rigorous approach at quantifying the amount of cortisol in these individuals, the body tends to compensate over the course of the day. And so we did not see an effect on those inflammation, mood, or sleep. All right, so the last study that I'm going to walk through is just wrapped up more recently in our laboratory just over this last year. And the lead scientist from my laboratory on this was David Alvarado. And then Tori Holthaus from Naman Khan's laboratory was the lead from their end. And we were specifically interested in this one in a specific fiber. So we were interested in the effects of soluble corn fiber, 18 grams per day, for 4 weeks on executive function, the microbiota, and connections between the gut-brain axis. This was a little bit larger study, so 42 adults that were older age. So their ages ranged from 50 to 63 years, had generally close to healthy BMI, just slightly in the overweight range. And then there was also a fiber intake criteria here where we were aiming for individuals that had a lower intake of fiber per day, more consistent with the general population. So the average fiber intake per 1,000 kilocalories for this group was 11.2 grams. And just for reference, what's recommended in the dietary guidelines is 14 grams per 1,000 kcals. So this group was closer to the standard American who, by and large, does not get adequate fiber per day. So same design as you've seen previously, where we had the 4-week intervention, a 4-week washout, and then the movement into the other group. So for these, for the bacteria that we saw a specific enrichment in for this study was actually in Parabacteroides. It was enriched after 4 weeks. What you're seeing here on the left is maltodextrin, our control, and on the right is soluble corn fiber, and these are the preconditions. The different stacked gray bars you'll see in the legend on the right, Bacteroides, Faecalibacterium, Parabacteroides is in orange, Latia, and Allostipes. These are just the 5 most abundant genera. And each bar represents each one of our study participants. And so in just a moment, I'll show the post conditions and just to highlight here an example of how much inter-individual variation that you can have in these microbiota results and why the continued growth and interest in the responder, non-responder, and personalized nutrition is very important. So what we found was at the end of the condition, the 4-week intervention, there was a main effect. So the group as a whole had an enrichment in Parabacteroides compared to the maltodextrin control group. So you can see each of those orange bars are going to increase in varying degrees. But you can certainly also acknowledge that some of these individuals started with basically no Parabacteroides. And so the ability to enrich those with the soluble corn fiber is going to be ablated because they don't have some of those key microbes that happen to be enriched with this study. And the enrichment in Parabacteroides was consistent with previous literature investigating soluble corn fiber. All right. So moving on we were interested in, okay, can we do a deeper dive? And so David actually took this one step further and did some in vitro experimentation to determine if Parabacteroides was actually able to ferment soluble corn fiber or if it was happening via cross-feeding. And so these different lines are showing the medium with either glucose in black, maltodextrin in gray, nothing, and then the orange with soluble corn fiber. And what you'll note is that Parabacteroides was able to ferment soluble corn fiber. It did have a longer lag phase, but then it was able to adapt and utilize it as a substrate. And so that was quite important for us to validate. And then one of the other things that we are interested in exploring was, as where we're at right now, is looking at some of these correlation matrix to determine if there are interactions between cognitive tasks, the microbiota, and microbiome metabolites. So first, just looking at the microbiota, the Parabacteroides was also positively correlated with greater concentrations of fecal short chain. So acetate and propionate, there was a positive correlation between those. And then when we looked at one of our cognitive tasks, which was an incongruent reaction time, which, and so the faster you can accurately respond to a stimulus, the better the performance there. We did find that with this, there was a positive, or there was a negative relationship with the congruent reaction time and acetate and propionate, meaning that individuals that had greater concentrations of these microbial fermentation end products were able to perform better in a congruent reaction task, which is a measure of executive function. And so we're still working through finalizing some of these analyses and hope to have the preprint out for this particular manuscript before the end of the year. But it is quite exciting to be able to, move these different research areas forward, look at the different types of fibers and prebiotics and probiotics and how they can have differential effects within a human clinical study. So hopefully what I've been able to share with you today as we think about this evidence hierarchy, and I know this is kind of a downer title here for this one, but we really still at this point have limited evidence for diet microbiota brain interactions within clinical, but there is a lot of evidence that diet supports brain health and that diet affects the microbiota. So it's promising and ripe for further investigation. I showed you some research on that we've been able to do, look and others on Mediterranean diet and the MIND diet, looking at dietary patterns, thinking about diet quality, where greater adherence was related to greater cognitive outcomes, and then also better mood. And then we talked about some different nutrients, particularly some different fibers, and then also the probiotics and the prebiotics, the work that we've done in our group. And so again, recognizing that, the research that is done within this field is going to, it is necessary to utilize, cross-sectional, longitudinal or cohort studies, intervention studies, as well as preclinical work and computational work so that we can piece this all apart. But we do want to make sure that we're being cognizant of the current state of the evidence because there tends to be a lot of hype still, it seems, in the gut-brain axis area as we've moved into the population, the general population as a whole. We are seeing some stronger evidence if you're looking at individuals with diseases. So, and so I'm really excited to be able to continue to work in this area as it evolves. So the last slide that I have here is just the key takeaways. Hopefully from this, you've been able to recognize that diet impacts the gut microbiota. I've also shown some data that diet impacts brain health. And kind of the key takeaway here is there is really exciting and growing evidence in the area of diet microbiota brain connections. But we'll continue to see this area evolve as we have more clinical research and more preclinical and more computational approaches to really understand what's going on in this. So thank you so much for your time and attention. And I'm really looking forward to the discussion during the panel very shortly.
Thanks so much, Dr. Holscher, for such a great talk. I think that we had two excellent topics that really kind of complemented each other, with Dr. Ritz talking about sort of molecular mechanisms in preclinical models, and then Dr. Holscher sharing with us population studies and clinical intervention studies. So I'm going to look at the Q&A now. at the questions. And then please, Dr. Holscher and Dr. Ritz, if you have questions for each other, feel free to just jump in and ask. Hopefully, yeah, we can kind of make this like a discussion. So yeah, feel free to jump in whenever. I'm looking at the Q&A now. Okay. Okay, I have one question. This first question for Dr. Ritz. Did you use an optimized extraction method to analyze the virome? And if so, do you expect the extraction method to affect your results?
Yeah, no, great question. I didn't really get into many of the methods to kind of keep it within time. But yeah, so we did use a optimized viral extraction method. It basically consisted of multiple levels of centrifugation, filtration, and then actually even dialysis to basically separate the specific size strata where we find viruses in the gut. And then from there, we reconcentrated them so that they would be at a higher abundance in the exact small gavage aliquots that we would then give to the mice. And the second part of the question, does it affect our results? Yes, of course, no matter how you do it, it's going to affect the results in some way. I actually saw one of the other questions that kind of got along this point as well, and it was whether we expect this to be more lytic or planktonic type bacteriophages versus lysogenic phages. And that's actually a very good point to bring up as well. We would not expect there to be many lysogens in this kind of fecal virome transplant. And that's because the way that we separated them by size was to remove all of the larger microbes. So all the bacteria, archaea, fungi were all separated. So the bacteriophages that were integrated inside of those cells would have also been removed. So the viruses that we did transfer from the earlier time point to the mouse during stress would have primarily been composed of the planktonic bacteriophages that were free in the fecal solution that we started with.
Great, thank you. I kind of had just a question, like as you were responding to, I mean, it seems like one of the big challenges is that we really haven't fully characterized the virome. So are there maybe like virome-wide, like, how are people approaching this problem? Is there some kind of like proteomics method to just like broadly characterize? Are people developing like a 16S equivalent or something like that for viruses or?
No, good question. Unfortunately, there is no conserved gene equivalent that we can use, and that's part of the difficulty of having these sequencing approaches for the virome itself. There are a lot of different databases being produced by a few different groups out there. So we worked closely with Colin Hill's group, also at APC Microbiome Ireland, and they have put together quite a comprehensive pipeline and series of papers where they've been able to look at the specific bacteriophages that we can find in the gut. But the primary difficulty of all this is that since there's no conserved gene to look at all of the different viruses, we can tell that there's viral sequences, but we can't have a good way of showing who all the individuals are. So it's kind of a painstaking approach now at this point, but every phage or virus needs to actually be characterized individually and added to new databases that are still being developed.
Okay, wow, yeah. Well, that's great that people are working on that.
Yeah.
Okay. This next question is for Dr. Holscher, and it says, you mentioned that dairy consumption was associated with lower stress on the DASS questionnaire. Was there any information on the type of dairy? I was thinking about how some molecules may be in all types of dairy, like conjugated linoleic acids, but others may actually be additives, like how vitamin D is often added to whole milk.
Yeah, so that's a great question. So for those cross-sectional and longitudinal studies, we were utilizing the validated food frequency questionnaires used by NHANES, so the diet history questionnaire, which queries intake over the last 30 days to a year. And so those are going to not be able to give you the level of sensitivity to determine, is it yogurt or milk. It's going to encompass things that fall within the dairy category, according to MyPlate, for example. But there is some really interesting research actually going on here at the University of Illinois, Jacob Allen's lab, in collaboration with Mike Miller here. They are very interested in what Jacob and Mike have done, and they recently published on this, and I think it gets to one of the other questions that maybe Sean or someone else asked in the chat, but there are different ferments that will have different microbial metabolites in them, and so some of those beneficial indoles, for example, that come from lactic acid bacteria will range in different types of yogurts versus sauerkraut versus, fermented carrots or, anything kombucha, anything you can find at the grocery store. And the fermented dairy products, the yogurts especially, can be really rich in those byproducts. And it's likely they are helping to underpin some of the positive immune responses that we've seen in the literature, right? With Dr. Cryan's group, Justin Sonnenberg's group, there's been some really nice research there looking at fermented foods, reduce inflammation, and some in the Cryan group, they had even the benefit in the mood, I believe was one of those. I'd have to go back to double check. But all this to say, depending on the tools you're using to assess diet, you can get different levels of sensitivity. So certainly when we do the interventions, we can feel, we're going to know specifically what was in the product, what was studied, if it's particularly having that impact. And so then just to briefly kind of wrap this all together with someone else had asked, what was the fermented milk we used, for example, in our fermented dairy study? That was actually a Lifeway kefir. So one that you can just buy in the grocery store every day was what we used in that particular one. And that certainly could vary versus other type of fermented milk beverages. They're really going to run the gauntlet, depending on what microbes are there and what the starting substrate is.
That's really interesting. I think especially the part where you mentioned that different fermented foods might have like different molecules, so maybe different postbiotics that you're actually consuming in the fermented food. I was curious, I think you primarily touched on pre and probiotic interventions. Are there any promising maybe postbiotic interventions? You mentioned the colonic butyrate, but
yeah, I would have to go back and look. I know, again, there's been some promising work in the fermented food section. And I think fermented foods is a really broad category, right? Technically, fermented foods includes, the coffee, probably most of us had this morning, chocolate, sourdough bread. But most of the most promising evidence that I've seen for fermented foods and health is going to be fermented dairy, so yogurts and the kefir beverages, but within the broader context of what those postbiotics might be. I haven't seen as much, but again, what I find fascinating is some of the microbial work they're doing here at Illinois with Mike Miller and Jacob Allen, where they're using the microbes in a way that they're manipulating the microbes to actually increase the amount of these fermented byproducts in the foods. So rather than providing a postbiotic, they're modifying the food in a way that will still be tasty and you can enjoy consuming it, and providing that as compared to just a standard yogurt that you would have in the grocery store. So we can think about potentially some of these next generation probiotics and how we may start to modify our food to make it more supportive of our microbiota and other health outcomes.
great, thank you. I have another question here for Dr. Ritz. Do you think there may be direct effects of the virome or phages on the host, such as activating the viruses themselves, or the phages activating host receptors independent of the bacteriome? What if you gave the FVT to stressed germ-free mice?
Yeah, no, that's another great question, and I think it's a valuable avenue for this type of research as well, because Classically, we consider the bacteriophages only indirectly affecting the host by infecting the bacteria and then changing their either population composition or the functionality of the bacteria themselves, then driving effects on the human or mammalian host. However, there actually have been some studies that have come out in recent years. The first one that comes to mind was one by done by June Brown's group, and they were looking at giving bacteriophages and different colitis. I believe it was ulcerative colitis, inflammatory bowel disease. And they actually had a really interesting finding that the bacteriophages themselves were capable of inducing TLR9 cascades. So there is some evidence that the bacteriophages can affect the host, but generally we kind of think of it more as affecting the bacteria and the bacteria then going on to affect the host.
Great, thank you. That's really interesting. I mean, I guess maybe we'll see if there are more kinds of interactions like that as we continue to discover and like characterize the viruses in the virome.
Yeah, no, absolutely. And that's going to be something that I think will be coming out probably in the next few years, seeing how the virome can have direct effects on the host. But it is interesting. I've seen some papers that have said that bacteriophages are able to kind of evade immune cell activation in the gut, and that they actually can be part of the innate immune defense against bacterial pathogens in the gut as well. But yes, so we'll absolutely see. And there's also differences whether you have an intact bacteriophage or a lysed bacteriophage, because they are ultimately viruses. And if you have a lysed bacteriophage, the different viral particles are capable of inducing inflammatory and immune responses in the host. So I think in a lot of cases, it just really depends on the context.
That makes sense. Okay. And then let's see. I have a question here for Dr. Holscher. Do you think SCFAs may be acting directly on the brain via systemic circulation, or might these reaction time effects be mediated by peripheral interactions with immune cells or enteric neurons?
Yeah, I think that's a really great question, and especially within the human models, it's really difficult to query that. I think with some of the animal models, building on that, it appears likely that the short chain fatty acids can directly affect the brain, and then they also have direct effects on immune cells in lots of different areas. And so with Dr. Verbecky's work that I showed that I found fascinating, she's previously done colonic enemas where they were providing the different short chains and they were labeled. And so they were able to track those in circulation and where they were going through different metabolic pathways and where they were ending up as triglycerides, for example, and processing through the liver. So again, these were in humans as we're measuring the systemic circulation through and how those short chain fatty acids get incorporated into different molecules. And so in that Verbecky paper that I alluded to there, they provided butyrate. And so they were able to look at the systemic circulation and how it entered. And then they also looked at fear and some other outcomes. And what they found, again, was a bit of a mixed response in those where, they had the butyrate, they were able to track it, but it wasn't an overly robust result with just the butyrate. And one of their conclusions, I believe, if I'm remembering correctly, was that they were thinking it may need to be the combination of the short chains instead of just providing the butyrate. So I would say the long and the short of it is we, again, have to really couple these clinical studies with the animal and the in vitro and the computational to be able to figure this all out, because even just thinking about humans and when we use stool and measure short chain fatty acids, my group is our participants, we do ask that they be able to bring their stool samples to the lab within 15 minutes of passing so that we can acidify those. And we use old rumen nutrition methodology where, just very carefully measuring those, acidifying them, using them on a dry matter basis. So we feel very confident in the amounts that we're able to measure. But then again, as we think about the take-home message there, it's the residual remaining amount of those short chains. We don't have the measure of what ended up in circulation, what's used in the cell. So I definitely think short chain fatty acids are a microbial metabolite that we should continue to focus on. But the human system is quite difficult to tease that apart in. And maybe Dr. Ritz has something to add on, because I think I saw short chains in one of your studies, or maybe it was in the background that you were alluding to. You could probably speak more eloquently to the connections in the animal models than I can.
No, I think you covered it quite well. But yeah, short chain fatty acids have been a huge portion of microbiome research at large. And we do have studies showing that short chain fatty acids can reduce the effects of stress in mice as well. And it's primarily looking at butyrate as like the key short chain fatty acid, but there are some data that show that acetate or even propionate can have some beneficial effects. But yeah, absolutely. Fermentation generating more short chain fatty acids, I think, is one of the key functions of the microbiota that needs to be both looked at and probably measured in different diseases and disorders to see if it's something that we can generate or supplement either in the diet or in some way to kind of reduce different effects, because butyrate itself has been shown to have large effects from everywhere on the immune system itself to even all the way to the brain and different HDAC inhibitors. So I do think that it's absolutely a key area of focus.
I guess maybe a question for both of you. What are the obstacles that we need to overcome, or how can we try to close that gap between preclinical studies and clinical intervention and observational studies?
I'll take the first crack at it, I suppose. There's so much to be done with microbiome research, and it's exciting to see where everything is going. But I think some of the key areas are defining specific mechanisms by which the microbiome interacts with the host, so that we can use that as different systems to treat therapeutically. I think assessing causality with the microbiome is also key, because there have been many studies that show associations and correlations between a specific microbial composition and a disease or disorder. But what we really need to show is whether the microbiome is causing the difference in the disease or disorder, or if it's a downstream result of the disease or disorder. Because I think we've seen cases go both ways. So being able to kind of assess causality both through in human studies with longitudinal type studies, so you can see the microbiome composition before a disease or disorder is triggered, and see if there's anything that kind of presets the disease. I think that's quite key. And then in different types of animal models, we're able to test a wider variety and more controlled environment to see if they drive these effects. And then I think in vitro studies are key for doing this more mechanistic work and seeing what specific receptors and ligands are really underpinning the entire processes.
Yeah, I think those are a lot of really great points, because again, since this really is such a newer field in a lot of ways, there are so many different areas we still need to flesh out. So kind of my favorite ones are going to be thinking about more dietary biomarkers and how we can more accurately and objectively assess diet so that we can more clearly connect it with any type of disease and any type of change in the microbiota. So that's definitely an area that I'm interested in. And then just to echo what Dr. Ritz said was these more mechanistic studies, so we can determine, is it a mediating or moderating effect of the microbiota, or is it independent? Because there's certainly going to be cases in all three of those boxes. And so coupling clinical studies with either preclinical models with transplantation or in vitro models, the computational approaches, using kind of this multifaceted approach can be really helpful. And then one of the other areas that I definitely see growing more and more, and I'm excited to also be involved in, is thinking about personalized nutrition and how we, with any area, the gut-brain axis or any type of health, but because we do have different microbial compositions within our body, some of us are going to be able to better utilize certain prebiotics, for example. So we've seen this for a long time with prebiotics. So FOS and GOS, for example. Individuals, if you don't have a lot of bifidobacteria already present within your microbiota, you're not going to have a bloom and an improvement in some of these downstream outcomes. And then in the study that we showed where we provided the soluble corn fiber, and David is really interested in investigating further how Parabacteroides may be a potentially a keystone species for breaking down the soluble corn fiber and contributing to those production of the metabolites, and how that might be connected with different cognitive outcomes. So again, it's a really exciting area to be in there. And I think a lot of partnerships with public-private partnerships can be really helpful too, as we think about high throughput approaches like SHINE models to test different probiotics with different, fecal ferments, for example. So it's a really great area. And so hopefully people that were tuned in today and listen to these later are feeling excited about all the research possibilities that we can all get to continue to contribute to over the coming years.
Yeah, thank you for that. I mean, I think something that's so cool about this sort of emerging research area is that it's interdisciplinary and requires a lot of different tools and approaches that really all need to be combined together. Yeah, I think we have a few more questions here. I have a question here for Dr. Ritz. This person asks, is there a sex and or age specific effect in the effect of FVT or the mediation of this effect?
Yeah, no, another good question. Unfortunately for this study, we did just test it in male C57 Black 6 mice. So we don't know whether there would be sex-dependent effects. But in many cases, you will see differences how males versus females process stress, and whether it can be something that goes down to the microbiome level. Generally, we will see differences in the microbiome across both sexes, and because of that, I would imagine that the fecal biome transplant would work similarly to how we saw in males, also in females. It would just probably be a different set of microbes in some contexts if they were found to be different.
Okay, and then kind of, I think, springboarding off of that, this is a question for both people. One challenge the microbiome brings up is the context specificity that it introduces. We all have unique microbiomes with unique complements of genes. So how much are we missing by focusing on developing population scale interventions instead of precision or personalized interventions? Might this be part of why the clinical results have been equivocal or inconsistent?
Yeah, and I can start on some of that. And I would certainly agree, right? The personalized nutrition and precision nutrition is really important. I'm a registered dietitian, so we work specifically with one-on-one individuals to come up with interventions that will work for them. And when we apply that to the microbiome area, we can think about how, again, baseline microbiome is likely to matter in the specific substrate you're introducing. So again, does the microbiome have the metabolic capacity to break that down so it can be effective? Or might there be other avenues that we can go? And then I also do think we likely, if we go back to old data sets and where the vast majority of the time we are looking at these means, there probably likely were studies where the intervention may have been effective in kind of these quote unquote responders versus non-responders. And in the prebiotic space, I do feel like that has been investigated a little bit more, where that was a more common term and individuals would try to investigate that. Sometimes you get a lot of pushback from during the peer review process, which is understandable because you do have to balance that with just data mining and p hacking. And so certainly I think there's opportunities to go back, look at different data sets, determine what characteristics might be underlying some of these responders and non-responders. And then you can use that information to go into other studies to either, pre-select a subtype that may be more likely to respond, and you can go in with those hypothesis testing. But certainly we can use microbiome research to as hypothesis generating. We just need to be transparent in our approach and be careful that we're not overstating the findings in that regard.
Yeah, no, I totally agree with Dr. Holscher's points there. I would just echo the thoughts that it's hard to know what you need to add to a system before knowing what's missing. So whether we're going to take some strain of lactobacillus or bifidobacterium might not be helpful in a lot of contexts, unless beforehand we know what we're trying to put there to increase the functional potential of those bugs. So that's why I also think combinations of using prebiotics and probiotics, and in some cases also postbiotics, so that we can give them the specific things that they need to then produce the metabolites and molecules that we are missing. And in a lot of cases, I think it just comes down to we need to know either what the composition is, what the functionality is, and how we can go about things. And in a lot of cases, we see that probiotics actually don't engraft all that well when they're taken by patients. And I think that's all the more reason why diet is so important in this, whether it's increasing your fermented foods or your fiber intake. There's a lot of different ways that you can kind of sculpt the microbiome, and perhaps in concert with different dietary regimens, you can then add different probiotics to kind of aid their engraftment into the gut and maybe have more stable effects downstream.
That I'm looking at, I think we have time for maybe like one or two more questions, but you mentioned like probiotic engraftment and perhaps maybe like dietary interventions may be more effective. And so somebody here is asking, many people have reservations about consuming dairy products, so they're of course not consuming fermented dairy either. So do we have any studies that explore any equivalent non-dairy food sources that are packed with the same nutraceutical properties? And so in addition to this question, I'm also kind of thinking about patients' compliance with the interventions, whether they be probiotic or dietary. Yeah, like what are good approach, like I guess what are some approaches that we can use to like get people on track with like a good diet that they also like enjoy eating?
Yeah, that's a $1 billion question. It's great, right? Like it's super important. Unfortunately, I mean, the average healthy eating index score, it's based out of 100. The average US adult is like 56, right? So we were just not meeting the standard general nutritional guidelines to support total health. And so in that, some of those, when we think specifically about the microbiota, is fiber-rich foods. So eating more fruits, vegetables, whole grains, nuts, and legumes. And I mean, trying to just even encourage people to do something simple like eating 5 fruits and vegetables per day can be some of the places, that dietitians will specifically work with people to improve. But yeah, it's very challenging because you have to also acknowledge that there are lots of different facets that come into this, right? Socioeconomic status, health disparities. It's, some of these foods, these fruits and vegetables, I mean, a container of raspberries is not cheap. They're a really great source of fiber, and phytochemicals, which are great for our health and also the microbes, and but they're expensive, right? So ultimately, as we think about the diet, it really has to go all the way up to like public policies and supporting initiatives so that people can afford to have access to healthful foods, and also the time to prepare them, and the education, right? Being able to know how to eat a head of broccoli in a way you enjoy it is not always easy, right? I didn't know that I actually enjoyed broccoli until my husband started making it in an air fryer with a little bit of olive oil, right? I won't eat it raw. I know it's super healthy, but I just have that bitter taste receptor and I just don't enjoy it. So those kind of educational approaches. So long story short, I mean, the dietitians play a really big role in this, nutrition educators, it's complicated. And as far as just really briefly to kind of add on with the probiotic question and the engraftment and different opportunities is what we know for that has been somewhat interesting with elderly individuals. If they consume a probiotic within a different context, we have seen subsistence for up to 6 weeks, that slower gastrointestinal transit time. And I think Jens Walter just had a nice study where they were trying to look at kind of those treatments and how long it would engraft with just one. So again, these studies are really important. But ultimately, when we're thinking about probiotics, I do always point people to like the US probiotic guide, because probiotics can be expensive. And if you need them, if someone asks me like, should I take a probiotic? I say, for what? And then they can go to like the US probiotic guide and see what the different level of evidence is for certain probiotics for certain health outcomes, because a lot of them can be useful for preventing antibiotic-associated diarrhea, but there's not a lot of strong evidence in that guide for mood, for example. And the Cryon group has, tried some probiotics in animals, and then when you try them in certain populations, it doesn't work. So I don't even remember what the original question was, but suffice it to say, nutrition is so important. It's so complicated, and I just love being able to work on multidisciplinary teams to try to solve these problems, because no one can do it alone.
Yeah, and I think those are all great points. Just back to maybe like the specific question of if you are lactose intolerant and you are seeing all these great effects by fermented dairy products, there are still a lot of fermented foods that are not milk-based. So whether it's going for a water-based kefir or a kombucha or a number of things like miso soup, sauerkraut, the list goes on and on of different fermented foods you can try that may all help. And for people that can't take the dairy products, I'd say point them in that direction first, perhaps. And yeah, as far as like the dietary side go, like one of the key things I think of as well is how important getting a diversity of different fibers is to produce a diverse microbiome. You won't get the same effects even if you eat a bushel of apples and you have a huge amount of fiber going into the system. It's probably going to help only a few different taxa, but the more broad, different dietary fiber intakes you can get, so different fruits, vegetables, like you're almost better off getting the wide variety and trying to work that into your weekly routine than you are just trying to up your total volume of these different fiber sources.
Right. Yeah, I totally agree. And I think we're pretty much out of time, but I think that's like a great spot to end. Totally. I think, yeah, we need these multidisciplinary approaches, ranging all the way from like molecular preclinical models to clinical scale models. And I think, as we continue to elucidate these different mechanisms by which like the gut microbiota might be affecting certain behavioral phenotypes, I think we also need to ultimately think about people's access to these kinds of treatments and interventions, and make sure that the access is equitable. So with that, we have a small break, and then we will have our session 3 on neurodegeneration. If you could all help me thank our 2 speakers, Dr. Holscher and Dr. Ritz again. And yeah, we'll see you after the break. Thanks.
Welcome back, everyone, to our third and final session of the symposium on the theme of neurodegeneration. I'm thrilled and honored to introduce Professor Priyanka Baloni, join us virtually from the School of Health and Science in Purdue University in West Lafayette, Indiana, and Dr. Yongwang Xu, joining us from the Genomic Medicine Institute in Cleveland Clinic, Ohio. Please continue to leave your questions and upvote the questions others have asked in the Q&A function and discussion during the Q&A session. And we'll have the discussion during the Q&A session. And don't forget to indicate which speaker the question was referred to. So our first speaker and my dear colleague and friend, Dr. Priyanka Baloni, is an assistant professor in the School of Health Sciences in Purdue University. Prior to joining Purdue, Priyanka was a senior research scientist at ISB in Seattle. Priyanka specializes in systems biology and its application to understanding complex health-related outcomes and specifically cognitive health. Her lab focuses on developing computational approaches to analyze and integrate multi-omic data, including genomics, transcriptomics, metabolomics, and proteomics, using in silico metabolic models and applying machine learning techniques to study the effect of gene-environment interactions on neurodegenerative diseases and other health-related outcomes. She will talk to us today about identifying host and microbiome-derived metabolic signatures associated with Alzheimer's. Priyanka, take it away.
Thank you so much, Noah, and thanks for the invite. Glad to be like a presenter in the Microbiome Symposium. I'll start sharing my screen. And let's see. Is it visible properly?
We see the presentation mode.
Oh.
There we go.
Is it? Okay, perfect. So thank you all. Thanks for joining us today for my talk, and thanks, Noah, for the kind introduction. So today I'll talk about like my study about studying the bile acid metabolism and how it is impacted in Alzheimer's disease. And before I jump into the talk, I just want to give like a brief overview of my past and current research work. So in the past, I was part of the Institute for Systems Biology for almost 6 years, and I moved to Purdue University 2 years ago. And my research focus was mostly on Alzheimer's disease and health, brain health. I also had my projects which were ongoing for cancer, pregnancy research to identify the metabolic markers that were associated with preterm birth. I also did my research on infectious disease as a PhD student and also as a postdoctoral fellow. I also worked on aging, gut microbiome analysis, and my current focus is on toxicology, specifically into studying how the gene-environment interaction occurs and how can we identify some of the neurotoxic toxic signatures. So my entire focus of research has been on metabolism, and I'll walk you through some of the research that I have done. So today I'll talk more about giving an overview of Alzheimer's disease, its application, and how we can use some of the in silico models, such as the genome-scale metabolic reconstruction, in predicting the disease pathophysiology and identifying, like one of the case study that I showcase here today is about studying how altered bile acid metabolism impacts the disease progression in Alzheimer's disease. So in order to bring everyone on the same page, I'll give an overview of Alzheimer's disease, as most of you would have heard the term that Alzheimer's disease is the leading cause of dementia. It is categorized as either early onset or late onset of Alzheimer's disease. And the characteristics of the Alzheimer's disease is basically the formation of beta amyloid plaques and the tau protein tangles, which build up inside the cells and are the leading cause of neuronal cell death. Currently, there are 2 FDA-approved drugs for the Alzheimer's treatment, and there are other drugs which are usually prescribed to the patients are for improving the cognition and other symptoms associated with the disease. So since Alzheimer's is a multimodal disease, there is a lot of ongoing research to understand how this disease progresses, because there are different different trajectories for individuals who have Alzheimer's disease, and especially like women are at a higher risk of getting the disease. And also like we are trying to identify the biomarkers for the disease that would make the diagnosis as well as the prognosis of the disease faster. So this is an overview of the physiological structure of the brain and neuron. What you see on the upper panel is the healthy brain. So you can see over here, like how the cerebral cortex as well as the healthy neurons look like. In case of Alzheimer's disease, what you observe is that there is a shrinkage of the cerebral cortex, as well as you find the deposition of proteins such as the tau tangles, as well as the A beta plaques in the neurons, which are the leading cause of the neuronal cell death. So these are the main differences that you observe between a healthy brain as well as an Alzheimer's disease brain. There are various risk factors which are associated with the disease, the primary ones being age and gender. And as I mentioned previously, women are at a higher risk for having Alzheimer's disease compared to the males. A lot of research has been focused on the genetic risk factors that are associated with Alzheimer's disease. Some of the studies have also focused on the environmental factors that lead to increased risk of Alzheimer's. Conditions such as obesity, diabetes have also been linked to Alzheimer's disease, in addition to cardiovascular diseases as well as head injuries. So there are various factors that are leading to like the increased risk for Alzheimer's disease. And the most predominant one that has been studied are the genetic risk factors associated with the disease condition. So what you see over here is a gene, the ApoE gene, which has been studied in detail because of its gain of toxic function as well as the potential loss of function. And it's a strong association for Alzheimer's disease. So it's one of the strongest genetic risk factor associated for the Alzheimer's disease condition. The ApoE gene is involved in various functions, including like if there is a presence of the E4 allele, it leads to increased brain atrophy, the neuronal toxicity, A beta aggregation, tangle formation, as well as the aberrant brain activity. Based on like the functionality of the protein, so ApoE leads to the production of the ApoE protein, which is involved in the transport of the lipoprotein, the fat-soluble vitamins, as well as cholesterol. And the statistics involved in the allele distribution is that there are 3 different isoforms, E2, E3, and the E4. And the worldwide frequency of these alleles vary. So the population of individuals who have the E2 allele is about 8%. E3 allele are the highest, they're about 78%. And the individuals with E4 allele are about 14%. An individual who has 1 copy of the E4 variant has 2 to 4 times higher risk for Alzheimer's disease compared to those who are E3 homozygotes. And individuals who have 2 copies of the E4 variant are 10 to 30 times higher risk for Alzheimer's compared to the E3 homozygotes. In contrast to all these, the E2 allele is associated with greater longevity as well as reduced age-associated cognitive decline. So there are different functions of these alleles and different functionalities associated with Alzheimer's disease. So currently, a lot of clinical trials that are ongoing and that are funded by NIA focus more on inflammation as well as amyloid. So you can see there are 2 different categories of clinical trials which are ongoing. Some of them are pharmacological, the others are non-pharmacological. Most of them focus on inflammation, amyloid, as well as the recent studies that have focused more on metabolic changes, as well as the bioenergetic changes associated with the disease condition. So a lot of focus is now being given to all these metabolic changes that can lead to the pathology of the disease. Some of the non-pharmacological interventions include exercise, neurostimulation, as well as cognitive training. So in order to get more detailed insights upon what kind of changes occur in the individual, in the disease condition, we need a ton of data that has to be collected from the population. So current efforts which are ongoing is collecting information from either the human or mouse or other model organisms that can be used for studying Alzheimer's. And we have currently like different consortia that have been built to study Alzheimer's, such as the AMP-AD, Model AD, and the other consortium. So AD Knowledge Portal hosts a lot of information related to the biospecimens that have been collected from the individuals, which include the data from brain, blood, iPSC cell lines, and others. And also, like, these have been processed to identify some of the omic-related changes, such as you have the transcriptomic data, metabolomics, proteomics data, as well as the SNP data for the individuals included in this study. In addition to like knowing all these multi-omic changes that take place in the individual, we are also looking into some of the neuroimaging changes that are associated with them. So these are the Alzheimer's Disease Neuroimaging Initiative has data related to individuals, especially the PET as well as the MRI scan that has been done on the individuals, so that you can look into how these gene-related changes can be correlated with all these neuroimaging changes that you observe. So using the data from that has been collected from the consortium as well as the neuroimaging data, what we are trying to do currently in my group is integrate all this information into genome scale metabolic reconstruction and identify some of the metabolic changes associated with the disease condition. So what you have learned in the past 2 days, as well as heard some of the talks about like genome scale metabolic modeling. So these genome scale metabolic models give us like a handy tool to assess, like based on the genotype of the individuals, what would be the predictive phenotype of them. And you can look into all these changes that happen at the biochemical level. So you can look at the reaction level changes. You can look at the genes involved in various reactions and how they implement all these changes in the organism. So the genome scale metabolic models are basically like biochemical information that has been like broken down into a mathematical format. And once you solve these models, you can predict what kind of changes occur at a system level. So these metabolic reconstructions are basically knowledge bases, which can give you information about different reactions. They can give you information about metabolites, genes, as well as different genes involved in different reactions, and how can you assess them. So you have the different knowledge bases that are currently available. What I have shown over here on the left panel is the human reconstruction. So this human one model is one of the comprehensive metabolic reconstruction for human that has about 13,000 reactions, 10,000 metabolites, and about 3,600 metabolic genes. On the right panel is a snapshot of what you can do if you have the organ-specific information. So you can build organ-specific metabolic networks and then assess the metabolic changes that are happening at an organ level. In addition to it, you can also build cell-specific models and then look at the cell-specific changes that are taking place in the organism. These genome scale models come with different functionalities, and you have different ways of integrating omics data with them. So one of the aspects here is that you can integrate omic constraints such as transcriptomic data, proteomic data, metabolomics data to understand how different conditions are affecting a metabolic state of the individual. You can also predict the regulatory changes, especially the transcription factors that are regulating all these metabolic changes in the organism. So you can apply all these regulatory constraints to it. In addition to that, you can also perform like in silico gene knockout as well as reaction knockout and predict like which genes are essential in the system. So all these functionalities are possible because we have different tools as well as functions available that can exploit these genome scale models and also integrate multi-omics data with them. So the work that I'm presenting today is about the collaborative effort and the result of the Alzheimer's Disease Metabolomics Consortium. And there are different centers involved in this consortium that have like focusing more upon the metabolic changes that occur in the Alzheimer's disease individual. So the main focus of this consortium has been to identify metabolites that are associated with cognitive impairment in Alzheimer's disease. And the basic readout that we have over here is to look at metabolites and how they are changing in different conditions. So the consortium has focused on different metabolic pathways, including like lipid metabolism, carbohydrate, amino acid metabolism, energy metabolism, which is linked to mitochondrial dysfunction in Alzheimer's disease. And we have also looked into the steroid and bile acid metabolism. So today I'll talk more about the bile acid and how we have identified some of the altered bile acid metabolism in Alzheimer's disease. So those who are interested in reading more about the work can refer to the paper that was published earlier, in which we used the postmortem brain transcriptome data from 7 different brain regions, which came from like 2,000 postmortem brain samples. And what we tried to do was, first of all, generate a brain region specific metabolic network, and then overlay the omics information that we had to identify how the bile acid as well as the cholesterol metabolism is impacted in Alzheimer's disease condition. So some of our findings led to the identification of transporters that were important and also highlighted the role of the gut-brain axis in Alzheimer's condition. So when I talk about bile acids, what are actually bile acids and how much do we know about them? So bile acids are the end products of cholesterol metabolism. And as you can see from the pathway diagram over here, so cholesterol clearance or the cholesterol efflux leads to the production of primary and secondary bile acids. You have 2 different pathways, the classical pathway as well as the alternative pathway that are involved in the production of bile acids. The primary bile acids are essentially synthesized in the liver, and the secondary bile acids are produced by the gut microbiome. So remember, these are the key terms, that the primary is produced by the host and the secondary by the gut microbiome. Some of the recent studies highlighted that bile acid profile changes in the Alzheimer's disease individual, which is associated with cognitive impairment. So bile acid profile has been studied not just in Alzheimer's disease conditions, but also in Parkinson's and other neurodegenerative disorders. A work that was published earlier by the collaborators, they performed the brain metabolomics analysis, and they identified that when you compare the metabolomics data in Alzheimer's individuals compared to the cognitively normal ones, there was a decrease in the primary bile acid level. So there was a decrease in the cholic acid level, and there was a corresponding increase in the secondary bile acids, such as the deoxycholic acid. So a difference in these bile acid levels was what led us to investigate further into what are the metabolic changes associated with the cholesterol as well as the bile acid metabolism, and how can we relate them with the cognitive decline in Alzheimer's disease. So in order to carry out the work, what we did was we collected data from 3 different studies, the Mayo Clinic, the ROSMAP study, as well as the Mount Sinai Brain Bank. And this study led to the collection of brain regions from 7 different parts. So we had the data from the temporal cortex, cerebellum, the prefrontal cortex, frontal pole, and the 3 gyruses. Using the transcriptome data, we were able to identify the genes which were involved in the cholesterol regulation, as well as transport and metabolism. And here you can see like the expression of cholesterol regulators such as SREBF1 and 2. They were expressed in all the brain regions. ApoE was expressed in different brain regions, as well as ABCA7, which is another cholesterol transporter. So we knew the expression of different cholesterol regulating genes in the brain of human. We also studied the bile acid related genes. So we looked into the bile acid receptors, bile acid transporters, as well as the bile acid synthesis genes. And an interesting finding that emerged from here was that FXR, which is a known receptor for binding with bile acid, was not expressed in the human brain. In contrast, we found the expression of PPARs, LXRs, and RXRs that were expressed in the brain of the individuals. So this led us to hypothesize that bile acid might be binding to other receptors in the brain that could lead to different functionality. Then we looked into the genes involved in the bile acid pathway and found that CYP7A1 and CYP8B1, which are the genes involved in the classical bile acid pathway, were not expressed. But in contrast, genes in the alternative bile acid pathway were expressed in the brain of the individuals. So this was suggestive that classical pathway of bile acid synthesis is less active than the alternative pathway in the brain. So from the analysis, we identified that the alternative genes in the alternative bile acid pathway were expressed. In addition to that, we also curated another pathway from literature, which was called as the neural cholesterol clearance pathway. And this was shown to have, the genes in this pathway were shown to have a higher expression. So this led us to look into details about different reactions that were important in the brain of these individuals. So for that, we constructed the genome scale metabolic network of the human brain regions using the data, as I mentioned earlier, from the ROSMAP, Mayo Clinic, as well as the Mount Sinai dataset. And we looked into the construction of 7 different brain networks that were constructed. And these were all region specific. At the end of it, we compared like which of the reactions in Alzheimer's versus the cognitively normal individuals showed a significant higher flux, and which of them were related to cholesterol as well as the bile acid pathway. So from our analysis, we were able to identify that certain bile acid transporters, especially the ones that were involved in the taurine transport, showed a significant higher flux in Alzheimer's compared to the cognitively normal individuals. And then we looked into the metabolomics data that was there from the blood of the same set of individuals. And from the metabolomics analysis, the levels of taurine were found to be higher in Alzheimer's compared to the cognitively normal ones. So increased transport of taurine across the blood-brain barrier could be responsible for the oxidative stress condition in the brain and responsible for the neuronal cell death. In addition to looking at just the transporters, we also looked into some of the secondary bile acids that were important. So in a gist, the metabolic networks of the brain regions were useful for predicting like differences in bile acid metabolism in Alzheimer's versus the cognitively normal individuals. So we wanted to explore further, like which of these genes were regulated by the transcription factors that could pinpoint like how this regulation happens. So using the brain transcriptional regulatory network that was built as an effort by Dr. Corey Funk and Paul Shannon, we were able to identify some of the transcription factors such as SREBF2, which regulated ABCA1. And ABCA1 was a bile acid metabolism gene, which was linked with, like, it was a candidate risk gene for the late onset Alzheimer's disease. STAT1 was a transcription factor regulating SLC6A6, which was a taurine transporter. CREB3L2 was a transcription factor regulating the CYP27A1, which was involved in the alternative bile acid pathway. And PO6F2 was involved in the regulation of EBP. So we were able to extrapolate the information from the metabolic networks to understand how their regulation is happening. When we published this study, we were like cited by someone on Twitter at that time, saying that one of the drug combinations that they have was having a combination of the tauroursodeoxycholic acid. So this is a secondary bile acid that has been that they were now planning to have like in combination for phase 2 clinical trials for Alzheimer's disease. So if you are able to supplement the individuals with Alzheimer's disease with some of these secondary bile acids, it can help in elevate the cognitive decline in the individual. So this was like a translational aspect of our study. So as I mentioned, like we had the metabolomics data from the postmortem brain samples, and the presence of secondary bile acids in the metabolomics data suggested that there was a possible role for the gut microbiome in Alzheimer's disease. So we wanted to like test whether these secondary bile acids are endogenously produced in the brain or they are transferred via the blood-brain barrier. The metagenomic study of the brain samples indicated that there was no microbiome data, microbes detected in the brain. And then in collaboration with Dr. Ines Thiele's group from NUI Galway, we were able to use the Agora model and identify which are the gut microbes that are able to produce the secondary bile acids. So amongst the gut microbes, they were able to pinpoint that Ruminococcus collinsella species as well as Ergothella species. They were the ones that were involved in the secondary bile acid production. So this gives us a hope that we can investigate further about some of the gut microbiome which are involved in the secondary bile acid production. Moving on, like we identified some of these bile acids. Now we wanted to look at the host aspect. So this is another story which we are currently working on publishing. So a graduate student in my group, Boyu Jiang, has developed the human colon epithelium metabolic reconstruction. And this is a reconstruction that was developed using the transcriptome data of the human colon epithelial cells, along with using different methods for building the draft reconstruction, and then testing the reconstruction by using two different case studies, the Crohn's disease as well as ulcerative colitis. So to tell a bit about the eye colon epithelium metabolic reconstruction. It has about 6,600 reactions, 4,000 metabolites, and about 2,000 metabolic genes. And using or integrating the transcriptome data from 2 different disease conditions, we were able to identify the altered metabolic pathway in these different IBD cases. And these were able to identify, help us identify some of the reactions that were important in case of the inflammatory group compared to the healthy or the control group. So look forward to hearing more about this work in a preprint that will be coming out soon. So some of the other ongoing work in my group is building the cell-specific metabolic reconstructions to identify how different cell types interact with each other and which are the cell types that are more important in regulating the changes that we are observing, especially the metabolic changes in case of Alzheimer's disease. Another aspect of the work is translating this information to identify condition as well as the sex-specific changes and looking for the markers that are specifically linked to all these sex-specific metabolic differences in Alzheimer's condition. We are also integrating the information about regulatory networks to identify transcription factors that are regulating all these changes. So a lot of work has gone into collecting a single cell as well as single nuclear transcriptome data, and then building the cell-specific models to identify these differences. So to summarize the work, as you would have in the past 2 days and today heard about a lot about genome scale metabolic reconstruction. So using in this present work, using the postmortem brain data, we were able to first of all generate the human brain region specific metabolic network. And then integrating the transcriptome data, we were able to identify that the alternative bile acid synthesis pathway is more active compared to the classical pathway in the brain. The presence of secondary bile acids in the metabolomics data is suggestive of the possible role of gut microbiome in Alzheimer's disease. From our study, we were able to identify some of the taurine transporters that play an important role, and as their levels were also higher in the Alzheimer's disease individual. So studying the gut microbes involved in bile acid metabolism will provide more insights into the AD pathophysiology. I would like to thank all the collaborators who were involved in this work. Special thanks to NIA for funding this work. And thank you, Sean and Noah, for the invitation to present in the ISB Virtual Microbiome Symposium. Thank you all for your attention, and looking forward to the panel discussion.
Thank you so much, Priyanka, for this talk, and we'll come back in the Q&A session a little later. I'd like to present our second speaker for this session, Dr. Yangwang Xu. Dr. Xu is a postdoctoral fellow at the Genomic Medicine Institute in Cleveland Clinic. He earned his PhD in drug design from the Shanghai Institute of Materia Medica, the Chinese Academy of Science. His research focuses on peripheral modulation of brain-related disorders by systematically leveraging AI-accelerated computational biophysics approaches combined with genetics and multiomics data analysis to identify peripheral chemical probes and therapeutic targets. Yangwang has published over 15 peer-reviewed papers, reviews, and patents, and he's currently working on developing an AI-based systems biology framework to reveal the potential of GPSR targets for gut microbial metabolites in the context of AD. He will talk to us today about his recent publication in Cell Reports and on the systematic multi-omics landscape between gut microbial metabolites and GPCR-OME in Alzheimer's disease. Please continue to deposit your questions in the Q&A session and state which speaker they refer to, and we will meet at the end of Yangwang's talk for the Q&A session. Thank you.
Can you hear me?
Yeah, all good.
Okay, cool. Can you see me by the video?
Yes, we see the
Okay.
slides fine.
Sounds great. Okay, thank you so much for your kind introduction, and thank you to all organizers for inviting me to give a talk. I'm very excited to share my recent research on transforming gut microbiome metabolites for the treatment of Alzheimer's disease by clarifying the G protein coupled receptors targets. So as Dr. Belloni introduced, the Alzheimer's disease, AD, is a neurodegenerative brain disorders accompanied by the accumulations of beta amyloid and tau protein pathologies, as well as the damage of neurons. The first AD patients was identified in 1902, and after over 120 years, the AD had been a leading cause of dementia, and with the aging population remarkable increase, it is anticipated that in 2060, over 13 million people in the United States and over 150 million people worldwide will have AD dementia. So despite a few decades efforts, unfortunately, over 99% of drugs have been failed in clinical trials. So there is still a challenge to developing AD treatment. So the right figure show that the current drug developing drugs are still targeting the traditional central nervous systems AD hallmarks, for example, the beta amyloid, tau, and neuroinflammations. It indeed works because in the last 2 years, 2 antibody drugs, lecanemab and donanemab, are approved by FDA. However, these drugs are only for early AD, not for late AD, and they are all antibody drugs. It means they are they have a higher they have a higher cost, and they are also targeting the beta amyloid, indicating the potential severe central side effects. So we are wondering, can we develop the peripheral small molecule therapeutics and drug targets for AD with less side effects and lower costs? So human gut microbiome may hold great promise for it because their diversity and functions have been playing critical roles in modulating AD pathologies through the gut-brain axis. For example, the gut microbiome toxemic diversity have been highly relevant to preclinical AD patients. And functionally, the fecal transplantation from the young mouse to the aged mouse could improve the inflammations of cognitive behaviors. And at the species levels, one recent study found that the administration of Bacteria's fragilis to the AD mouse model could increase the A-beta levels. And at a metabolite level, gut microbiome metabolites their valeribatine could reduce the age-related cognitive declines in mouse model as well. So these extensive studies suggested that it is promising to develop potential gut microbiota targeted therapies for AD prevention of treatment. To this end, the key is to understand the gut microbiota host interactions. The function of gut microbiota largely rely on their produced or synthesized metabolites. These metabolites serve as the messengers to act on the immune cells or vagus nerves in the gut, or act on the neuron cells in the brain directly by penetrating the blood-brain barriers. Some metabolites, especially gut microbiome metabolites, have been as an effective approaches for the disease treatment. For example, the short-chain fatty acid for the type 2 diabetes, and microbiome for the thrombosis. So here is a question, how these metabolites contribute to the disease pathogenesis, or what is their host receptors mediate their functional effects. For these questions here, in our studies, we focused on the G protein coupled receptors, GPCRs, because the GPCRs have been reported to selectively expressed in the enteric cells as well as the brain cells. And some classical GPCRs have been activated by the gut microbiome metabolites. For example, short chain fatty acid can activate the free fatty acid receptors. In recent years, some dark GPCRs, for example, GPR35 and GPR84, have been activated by the gut microbiome metabolites as well. These studies suggest that the GPCRs may be a large target reservoirs for the gut microbiome metabolites. In fact, the GPCRs constitute the largest memory receptors consisting of around 800 members. Of these, over 100 of GPCRs are still have little knowledge about their biological functions. And they are called orphan GPCRs or dark GPCRs. These GPCRs are expected to discover novel binding molecules. Furthermore, GPCRs are also an important drug targets, which account for about 30% FDA-approved drugs. So GPCRs, especially dark GPCRs, may be potential AD drug targets. In our studies, we aim to answer the following questions. Can we review the potential AD therapeutics from microbiome metabolites, GPCR molecular relationships by using structure-based computational approaches and machine learning models? And can we clarify the associations between the gut microbiome metabolites and the AD pathologies by systematically investigating the interactome analysis combined with the AD genetics and microbiomics evidence? To answer these questions, we developed a systems biology framework, which contains three parts. The first one is leveraging the microbiomics and the genetics analysis to prioritize the AD-associated GPCRs. And secondly, we're leveraging the molecular docking and the machine learning models to predict the 3D structure-based metabolites GPCR interactions. And finally, we experimentally validating our predictions using the AD patient iPSC cells derived neurons. Okay, so firstly, we collected 335 metabolites from 178 human bacteria strains in vitro, which is reported in a 2021 Nature paper. Here, we only focus on the human-sourced microbiome metabolites, and we removed the metabolites sourced from the colonized mice. And for the structural models, we collected around 2,000 models, including crystal structures, crystal structure models, homology models, as well as the AlphaFold2 models. It's worth to mention that the AlphaFold2 has demonstrated its ability to for the molecule screening. A recent science paper reported that a science paper reported that the millions of molecules was screening against the AlphaFold2 structures. They achieved a high hit rate for the AlphaFold2 structures compared to the crystal structures. In our studies, we also compared the alpha photostructures and create structures. We found that the alpha photostructures have a small transmembrane root mean square deviations. It indicated that alpha photostructures also have a high confidence transmembrane helix regions. So as we mentioned, firstly, we prioritize the AD-associated GPCRs. Firstly, we use the Mendelian randomization analysis. Mendelian randomization is a bioinformatics approaches that use the genetic variations to address the causal questions about how the modifiable exposures affect influence the different outcomes. Here, the gene expression is the exposures and AD pathology is the outcomes. Through the MR analysis, by using 3 AD-GWAS datasets and 4 EQTL datasets, we found the 6 GPCRs were significantly associated with AD. For example, the upregulation of TAS260 receptors is associated with reduced risk of AD. And next, we investigated the GPCR, whether the GPCR are also differentially expressed in the bulk transcriptomics and proteomics datasets. The right figure showed the GPCR phylogenetic trees and the number of datasets in which the GPCR are differentially expressed, shown in the outer stack bar. By the transcriptomics and the proteomics analysis, we found 10 GPCRs are differentially expressed in at least 5 AD mitomics datasets. Here we showed the top 4 GPCRs, including GPR84, CX3CR1, CXCR4, and GPR34 in 3D structures. And next, we predict the GANT metabolites and the GPCR interactome. Firstly, we identified their potential druggable pockets, including orthosteric pockets and allosteric pocket. The allosteric pocket indicated the pocket that with a new mechanism of actions. And next, we employed the molecular docking methods to predict their interactions. Compared with traditional high throughput screening, this method have a lower cost, a high efficiency, and a large chemical species. However, the limitation is there is a lower accuracy. To overcome it, we built 12 machine learning models to improve its performance, including decision trees, SVM, KNN, and other models. By comparing Pearson correlations, we found that the actual trees algorithm outperformed other machine learning models on the test datasets, benchmark datasets, as well as the traditional docking approaches. We named it as a GPCR ML score. Based on this score, we predict the GANT metabolized GPCR interactome, which including 1.09 million pairs connecting 408 human GPCRs and 335 GANT metabolites. And by the pair numbers and the category analysis, we found that the GPCRs are likely regulated by the lipid and lipid-like metabolites, particularly for the orphan GPCRs. And then we mapping our AD-associated GPCRs into the interaction network, and we've identified 220 interaction pairs, which is AD-specific pairs. And of this, the orphan GPCRs comprise the most proportions. Based on the AD evidence numbers, the right heat map showed the top metabolized AD GPCR pairs for 16 AD associated GPCRs connecting with 27 metabolites. The GALR1 and the FPR1, two GPCR receptors, are supported both genetics and multiomics evidence. And these two receptors are predicted to interact with both lipid and lipid metabolites. Since the microglia is also pathologically associated with pathologies via mediating neuroinflammations, so we also inspect the expression GPCRs that is differentially expressed in the disease-associated microglia. By the single nuclear RNA sequencing analysis, we identified 8 GPCRs were significantly differentially expressed in disease-associated microglia. For example, GPR34 and P2RY6 were differentially expressed in both A-beta-related microglia and tau-related microglia. Of this, GPR34 were found to pair with acid, which have been found to alleviate neuroinflammation in microglia. C3AR is a well-defined immune modulator have been found involved in the AD neuroinflammations and the tau pathologies. We predict that C3AR can be interact with 4 gamma metabolites. Of this, agmatin is a metabolite that have been shown protective effects from the neuroinflammations in AD mice. Okay, next we experimentally validate our prepared predict the pairs between the gamma metabolites and the AD-associated GPCRs. Firstly, we inspected the GPR84 because it has the most multiomics evidence. We inspected the top 50 metabolites of the GPR84. For example, the tricasein, tridecanoic acid, and S-glutamine. Particularly, the tridecanoic acid have been reported to activate the GPR84 with the EC50 of 48 micromolar. So that's the way we inspected the functional activity of tricasein of GPR84 via CMP assays in the HEK 293 cells. As a result, we found that these metabolites can activate the GPR84 with the EC50 of 32 micromolar, which is comparable to the reported metabolites tridecanoic acid. Next, we want to inspect the gamma metabolites from some AD-associated gamma microbiota. Here, we analyzed the metabolites abundance of 2 AD-associated gamma microbiota, including Eubacterium rectale and Bacteroides fragilis. We found that 2 metabolites, agmatin and tricasein, are 2 most abundant metabolites in the Bacteroides fragilis. So next, we use the AD patient iPSC derived neurons to inspect their functional associations between the GANA metabolites and AD-associated GPCRs. As we mentioned before, C3R is GPCR candidate, the top GPCR candidate for the agmatine. So we examined the effect, the functional effect of agmatine on the C3R, and we found that the agmatine could significantly reduce the C3R levels at 50 micromolar and 100 micromolar. We also found that the agmatine could significantly reduce the level of p-tau, including p-tau 181 and p-tau 205, in the iPSC-derived neurons as well. Furthermore, we also checked the genus Ruminococcus, a genus associated with AD as well. And we found the GANA microbiome metabolized phenylethylamine have a higher abundance in this genus. Based on our GPCR ML score, we found its top GPCR candidate is orphan GPCR GPR 153. Similarly, we also demonstrated that this metabolites can significantly reduce the tau hyperphosphorylations in a dose-dependent manner. So these experimental findings demonstrated that our GPCR ML score could significantly predict the functional GANA microbiota metabolites GPCR pairs where reducing the tau expressions in AD. So in summary, combining the machine learning and multi-omics analysis, we identified the molecular relationships between the GANA microbiota and the GPCR families in AD. This study may offer systems biology resources to identify the microbiome targeted therapies where targeting GPCRs for treating AD and other diseases if broadly applied. In these studies, we used the machine learning models to predict the over 1 million GANA microbiome metabolite GPCR pairs. And then we leveraging the genetics and multi-omics analysis to identify the AD-associated GPCRs or likely causal GPCRs. And by experimental validation, we found that the agmatine could reduce the level of C3R and the p-tau levels in patient iPSC-derived neurons. And similarly, we also found that the phenylethylamine could reduce the tau hyperphosphorylations in AD patient iPSC-derived neurons as well. So our work also have some limitations. For example, more experimental validations and deep mechanistic observations are also needed in the future. And our studies did not consider the pharmacokinetics of metabolites. For example, the blood-brain barrier permeability of metabolites, which may largely limit the efficacy of metabolites in the brain. And further targeted engagement experiments, for example, the knock-in or knockout, are also expected to confirm their specific activity of agmatine on C3R and other potential targets. We are still working on the GANA microbiota derived based therapeutics for AD by using artificial intelligence models. For example, the large language models to improve the metabolites protein interactions. Not only GPCRs, we are also working on the whole proteome target identification platform, including other proteins, for example, enzymes, nuclear receptors, and others. Not only the metabolites and protein interactions, we also working on the protein-protein interaction predictions by using AI models and metagenomics analysis. So finally, I'd like to thank my supervisor, Feixun Chen, and my colleagues for their help on the metaomics analysis as well as the experimental validations. I also thank the NIH funding supports. By the way, our lab, Chen Lab, Chen Lab is focusing on AI advanced network medicines for the drug discovery and target identifications for the neurodegenerative disorders. Our lab have built several comprehensive databases. For example, for the single cells, for the metaomics data sets, for the drugs in the clinical trials, and also for the potential targets, as well as my quantum GPCR interactions. So here I just show their barcode, and be free to scan this barcode to explore more information for your own research. So thank you so much for your attention.
Thank you so much to both speaker, Yang Wang and Priyanka. You're welcome to join us now for the Q&A session. Yeah, there you are. So again, thank you so much, both of you. I would like to start with a question to kind of both of you, I guess. Priyanka, you talked about ApoE, and we know it has a role in lipid shuttling. We know ApoE 2 and 4 differentially bind lipids, differentially carry lipids, and so on and so forth. So did you examine in any of your analysis the ApoE status of the AD cases, and whether that's kind of had a metabolic effect or effects on the metabolic fluxes changes that you observed? And I guess the same question for you, Yang Wang.
So yeah, that's an interesting question, Noah. And like, I'll try to pinpoint first of all that the study that we had in hand, the samples that we had in hand, had a lesser proportion of individuals who were ApoE4 homozygous. There were more of them who were ApoE3 and 4 and 3 and 3. So that's why like our study was more biased towards those who were heterozygous. And you can see the differences between them, especially like when I looked at the ApoE levels, like based on the ApoE stratified data, you could see that the bile acid differences were there for the ApoE4 E4 individuals compared to E3 E3 individuals. So you can see the genotype having an effect on the bile acid levels, but like we need like some more data to compare with whether the E4 homozygotes had higher bile acid levels or not. So yeah.
In the other direction, have you seen lesser effect or similar effect in ApoE2 if you had any?
Again, there were like very few individuals with E2, which were E2 homozygotes, so we couldn't assess and like find statistical differences between them. But having a cohort which is like which has all these mixed genotype of individuals, that would be something that we can investigate.
Wang, do you have any to add from your side?
Yeah, I just would like to add that from the structures perspective, the ApoE 3 or ApoE4 only have 1 or 2 residue mutations, right? And however, the tough question is that it's very hard to target it. It has this disorder structures, and that's why we want to explore more druggable targets, right? So So in ApoE, in our lab, we also study the structures of ApoE, and try to find some potential pockets from the ApoE4 or ApoE3 by using molecular dynamic simulations. It's very long, about 500 microseconds. However, we still cannot get some clues from this structural analysis.
Another question is to both of you, I guess, is both of you are looking at brain samples and brain cells, and I was wondering if you can comment about what you think is happening differently between the brain and the periphery, and whether we would be able to find the signals, for example, that you detect, Priyanka, in the periphery, if you ever looked at these data. And there was, I think, a related question in the chat, which I think you referred to, which is how do secondary bile acids make their way into the gut lumen through the BBB? But the interesting part of this question, I think, is that do you think that once the BBB becomes leakier as the disease progresses or with age, do we see an increased entrance of secondary bile acids into the brain and a larger effect, do you think?
Yeah, so to answer your question, like first of all, like the bile acid levels, they change in the individual, and we also like wanted to investigate whether they like cross the blood-brain barrier because it's leaky in the Alzheimer's individual or not, or whether it's age-related. So yeah, like a leaky blood-brain barrier does contribute towards the transport of bile acids. And it's like in the chat, as it's mentioned, it's more of an active transport that is happening because you require solute carrier proteins for transporting these bile acids to the brain. So it's an active transport that is happening. Regarding to your like, if you can repeat your first question, Noah, you were looking for which ones?
The differences between blood and periphery in terms of the signal that you think you're going to see.
So again, like to make everyone aware, so the brain data came like from the postmortem individuals. So what we are observing are after like the disease has set in and caused the death in the individual. For the blood, we have like a longitudinal data, which is currently available. And that's what we are trying to assess, whether like we can extrapolate the onset of the disease and then make predictions based on it. But the brain data is comprehensively post-mortem. So whatever we are observing is what has been like led to the disease or the death of the individual eventually. So it's not conclusive.
I would make a suggestion. The paper we just pre-printed yesterday together. Like you could look at people that are at higher risk for Alzheimer's disease to see if you see those like secondary bile acids higher for them. For example, that could be like indicative that this is somewhat not just reflecting the disease, but rather maybe causative for the risk.
Yeah, and we did find some of these signatures in the blood samples that were analyzed, especially like the blood metabolomics data that indicated that individuals with a higher risk have higher levels of some of these secondary bile acids.
Yeah, awesome. I wanted to ask you, Dr. Xu, just something I was curious about. So you related some of the mediating metabolites and microbial metabolites, which I thought was very interesting the way you extracted them. But I was wondering if you looked into whether any of them are diet derived.
Oh, you mean diet derived metabolites?
Yeah.
Oh, yeah, it also is very interesting topic. We are also, not only focused on the gut metabolites, we also collected, billions of metabolites derived from the food or diets. We can do together, we can predict their targets together by using the combinational approaches. And actually, we are working on that, and it's also interesting. However, the problem is that there's some cross talk with bacteria and food and even the human derived metabolites. The problem is that we cannot identify their source, right? So here is another question: is the concentrations of these metabolites? How much do we need, and which one should we more focus on? So we can do that, of course, but there are a lot of other questions we should consider about.
Yeah, I think if you join the rest of the course, like yesterday, when they showed metabolic models, maybe that could help you in kind of connecting and trying to see if you can simulate optimization of certain metabolites and target it. Do you want to target? Okay, so now let's go to a question from the chat. Priyanka, for you, do you know of any evidence that the secondary bile acid producers are differentially abundant in the guts to AD patients versus unmatched healthy controlled? Age-matched, sorry, healthy controls.
Yes, so that's a very interesting question, and thanks, Sean, for asking it. So I know, based on some of the recently published work, especially looking into the gut microbiome of the individuals with Alzheimer's disease, they have shown that there's a decreased proportion of some of the microbiome, such as those belonging to the Blottia and the Orgatella species. So they are decreased in the Alzheimer's individual, indicating that those who have a lower proportion of these gut microbiome are at a higher risk for having Alzheimer's disease, because they are somehow correlated with neuroinflammation also, like increased neuroinflammation. And I would just like to point out for the audience that the Alzheimer's disease gut microbiome consortium is currently collecting a lot of data, which is specifically focused on studying the gut microbiome and the effect of diet and other things which are related to all these things actually relate to the progression of the disease. So hopefully the consortium will have much more interesting findings that can report which of these gut microbiome are actually involved in the disease progression.
Dr. Xu, we have a question for you. Have you looked at the tissue-specific expression of the AD-associated GPCRs, and are any of them expressed in the gut epithelium?
Oh, in this study, we only focus on the brain tissues. I did check other tissues, and for example, we use the metaomics data sets. There are most of them from the human, cortex, brain regions. Yeah, that's a good suggestion. So I can also, check them. Yeah, thank you.
So, Priyanka, the next question is for you to you from Alisa. The question is, if I'm understanding correctly, taurine itself is neuroprotective, but taurine conjugated with lipocholic acid is predicted to be cytotoxic, and AD patients have a higher serum taurine and increased taurine transport activity across the blood-brain barriers. So then, is the hypothesis that microbial bile acid metabolism is decreasing the available taurine bile acids to be transported into the brain and increasing the amount of secondary bile acid in the brain, which may somehow shift the brain bile acid metabolism towards the alternative pathway, leading to more oxidative stress?
Yeah, so thanks, Alicia, for the question. And yeah, that's an interesting observation that you had. I also agree to it, because we have observed that the levels of taurine are in the serum of the individuals with Alzheimer's is higher compared to that in the brain, and this is indicative of the gut microbiome probably derivatizing the taurine so that it gets conjugated to the secondary bile acids. What we are currently not sure about is do the levels remain constantly higher in the Alzheimer's patient, or is there a fluctuation that might lead to like a different observation? So we need some more data to like substantiate the hypothesis currently, but like the transport of taurine has emerged from different studies, including ours, that it's one of the important factors that we need to focus on, especially when we are looking for the levels of secondary bile acids in the brain.
Thank you. The next question for Dr. Xu is just a small question. Maybe you can clarify. On one of the slides, you show like comparison between differential expression of GPCRs versus their AD associations, if you remember, right? And there were 10 that were, I think, differentially expressed and 6 that were AD associated. So I was wondering what were the 4 that were differentially expressed but not AD associated, and how would you explain that?
Oh yeah. Through the Mendelian randomization analysis, we identified 6 GPCRs may likely causal are related to the AD, and by multiomics analysis, we identify 10 GPCRs that are differentially expressed at least 5 multiomics datasets. So we combine them, 6 and plus 10, and then we use prioritize the interactions between these 16 AD associated GPCRs and explore more detailed analysis. Yeah. So because in our lab we previously compiled the 88 multiomics datasets, which have been published, and we have a database called GPS. Yeah, and so we based on this datasets to check the differentially expressed GPCRs, from the bulk transcriptomics or proteomics lab perspective. Yeah. Is that clear?
Yes. So the next question is for both speakers from Sean. I think Sean may be referring to a recent publication from his lab. Chronic constipation is a common comorbidity in neurodegenerative disorders. Actually, no, that's not but often arising years before diagnosis with dementia. But do you know if the organisms and metabolites that you are seeing associated with Alzheimer's disease show similar association with constipation?
I can speak to it. So I know that like one of the phenotype we are looking for is constipation, but frankly speaking, I haven't looked into specific metabolites as well as organisms that have been associated with constipation and then in the future leading to neurodegenerative diseases. But that would be an interesting area to investigate. Since like I'm sure like there should be data available to look for all these different changes in the individual.
Yeah.
Yeah, it's a very interesting question. As things that go into this topic. Yeah.
Okay. So Dr. Xu, do you think that GPCRs that do not show differential expression, for example, that may be constitutively expressed, may still be functionally related to Alzheimer's disease? Would activation assays or screens be more informative there?
Yeah, it's a really great question. So gene differential expression only so that there the associations between the GPCRs and AD, right? And it's I would say if there is a expression relationship, say maybe have a possible increase the probability to probability for the relationships between the gut metabolites and AD. However, I think it's not a a must or necessary to be connected by the expression and the AD pathologies. Yeah.
So the next question I wanted to ask actually Dr. Xu, you mentioned at your summary that you're now leveraging foundational language models, and I wasn't sure what you meant and how this could fit in here. If you can clarify and tell us a little bit more about that.
Yeah, because large language model showed a great promise to identify the interactions between small molecules and the proteins. And currently, we only focus on the machine learning models. However, there are many pre-trained large language models can be also employed to improve the accuracy for the predictions, right? So one one direction we are focusing on is the small molecule protein interaction predictions, and another direction is that we are focusing on the protein-protein interaction predictions combined with the metagenomics data sets. Yeah.
So how would the LLMs be used for that? Are you extracting the interactions from the literature using LLMs, or?
No, our goal is to build a systematic protein structure database, including all the AD-associated gut microbiota. For example, the Bacteroid fragilis I mentioned before. We may explore their protein sequences for all the protein sequences of these bacteria, and then we build their structure models for each proteins and build this structure metagenomics database, and then we predict the protein-protein interactions between the microbiome proteins and the host proteins, and using the AI models. Yeah, this is our ongoing project.
Next question is for you, Priyanka. Is your gut colon epithelium model able to integrate microbial metabolites?
Yeah, and that would be a great question, like for Sean to answer also, because we have currently got the host part of the metabolic reconstruction, since we are looking into the host colon epithelial cells. But our ongoing effort carried out by Nick Berman is about like integrating the gut microbiome with the host metabolic network. So yes, we will have the capability of integrating both the gut microbiome and the host system. But stay tuned for the work.
Next question is for Dr. Xu. There is a hypothesis that there are many conjugated bile acid receptors binding interactions that remain undiscovered. Were newly discovered classes of bile acids included in the ML model? And was there predicted activity between them in the orphan GPCR?
Yes, because we build a systems framework to predict all the non-olfactory GPCR receptors, including 408 GPCRs, which will include the bile acid receptors as well, and we predict the interactions between metabolites and these all the GPCRs. So this project we only take the AD as an example to show our power of frameworks, and just the combine the AD multiomics data sets and genetic data sets to prioritize the AD associated GP cells. And for other metabolites receptors, we also give our predictions, and all the results have been deposited in the database. I think there will be some interesting pairs, and researchers can be free to access to these predictions. Yeah.
And can I ask a follow-up question to Dr. Shu?
Yeah, sure.
Yes, go ahead.
Yeah. So I was like, since we know that immunometabolism is one of the key drivers for like AD pathology, so I was like very curious to know, can your models predict whether immune changes are the causal mechanism leading to metabolic changes, or is it metabolic changes that lead to the immune regulation? Can we predict it from your models?
Yeah, I think so, because but we need the data, we have to have the training data and predict the outcome, whether they're the immune regulations or other symptoms or other pathologies. Yeah, if we have a large training data, we can do that, of course. And we can use not only the machine learning models and other AI models to do that. Yeah.
That would be great.
Yeah, if you have these data, we can collaborate for it.
Yeah, I'm sure like the AD knowledge portal might have like the data that you are trying to look for, since they have huge cohort of individuals in that in their studies.
Okay.
Great. The next question is to both from Sean. It kind of relates to our previous discussion on brain versus periphery. So Sean is extending that to tissue. So you're both focusing, the question is, you're both focusing a lot on the brain itself, but do you think that microbe-host interactions in distal organs, tissues, may be driving some of these neurodegenerative phenotypes, for example, gut tissues, kidney, liver, etc.? So what measurements from the other sites would you like to take to better understand the AD phenotypes?
I can begin by answering it. So that's a great question, Sean. And one of the recent grants that we submitted was specifically focusing on liver, because we are trying to connect the liver, gut, and the brain axis together to understand how these are interconnected and are responsible for the disease progression. So we have like evidence which shows from different human-based cohort studies that the liver, especially like the diseased liver, leads to the production of altered bile acids, which eventually like cause an effect in the brain, and that is what we are trying to investigate. So yeah, liver is one of the key organs that we are studying. I'm sure like there would be other studies that have looked into kidney and others. So this would be interesting to study. The only problem here is that for the liver, we can only look for the liver enzymes that could be used for like correlation-based analysis. But getting like samples or the biopsy samples is one of the most challenging things. So if we have the ability or the funding to like look for biopsy samples and then connect them to like the progression of Alzheimer's disease, that would be like a tremendous work that can contribute to the field.
Yeah, I totally agree. And I think it's also a matter of data sets, because most of there is still a lack of data sets about the how the AD pathology affect the gut or peripheral organisms. So if we have enough like multiomics data sets or other genetics data sets, I think it will be helpful to infer the relationship between the peripheral systems and the brain systems.
So the next question is thank you both, is for you, Priyanka. So you mentioned that you're connecting proteomics and metabolomics data, but the proteomics data are limited to the ones that you have represented in your knowledge base. Is that correct?
Yes.
I'm wondering if there are other ways to connect, kind of to cast a wider net in the proteomic space, to connect other proteomic species in like a data-driven manner beyond the enzymes.
So beyond the enzymes, the other proteins that show like having an impact on the system are the transporter proteins. So if we have information of like, other than the enzymes, if we have information of how these transporter proteins levels are expressed in the individuals, that could give us a clue about the transport of different metabolites that are occurring and can help us pinpoint the specific reactions. So the proteomics data, like if it can be expanded to transporter proteins or the signaling proteins eventually, that would be somehow like connecting to all these inflammatory changes that we are trying to look for.
And translational modifications, for example, has that made its way into the knowledge models?
Currently not.
Currently not.
But hopefully we can integrate those information.
Thank you. The next question is for both, but I think maybe Priyanka, you are closer to that, as if there is a diet that could enable breakdown of cholesterol into bile acids instead of using statins.
So I wouldn't say like a diet that could enable the breakdown, but I would specifically say there are enzymes which are involved in the breakdown of cholesterol to bile acids. Like bile acids are usually produced at a higher level because they are required for the emulsification of the fats. So bile acids are produced at a higher level when you ingest like a high fat diet and other conditions. But I am not aware of a specific diet. I'm sure like the MIND study or some other pointer study would pinpoint which diet would be like useful for converting it. But I can say for sure like there are enzymes involved in the conversion of cholesterol to bile acids.
And so we are almost out of time, and I would like to pose the final question to you. Age is as the foundation of a lot of the things that you're seeing, and the highest risk factor for AD as well, and also affects a lot of the processes like mitochondrial function, energy metabolism, and so forth. A lot of processes at the base of what you're pinpointing. So I'm wondering if in any of your analysis you kind of factored age into this. I know, Priyanka, you had postmortem, but not necessarily all of them were very old. If you saw any effects of age on any of the outcomes and the associations that you were discovering, likewise you, Yang Wang.
So I did like look into the like when I collected the age of the individuals, I tried to look for age specific differences. But since most of the samples were 65 and older, we couldn't like identify significant changes that were with respect to the age. So if we have like, as I mentioned, Alzheimer's can be early onset or late onset. If we have individuals who had early onset cases, that could help us factor in like the age specific differences.
Would you like to add anything, Wang?
Yeah, but because I think the regulations of kind of metabolites or other environmental factors is how they contribute to the brain disorders, and I truly believe that it's because the molecular regulations, where the small molecules or their proteins, that is the basis. So about the other factors, I really I have no ideas for that. Yeah.
Okay. Thank you both. We are out of time for this session. I'd like to conclude the Q&A session with another warm thank you to Dr. Baloni and Dr. Xu. And thank you all for joining us for our third session, as well as a round of virtual thank yous for all of our fantastic speakers in this symposium. With that, I will return the floor to Dr. Gibbons for his closing remarks.
All right. Well, what an incredible day of talks. And great questions from you, the audience. Thanks so much for your engagement throughout the day and throughout the course. We hope you enjoyed our 2024 Virtual Microbiome series. Thanks again to our sponsors, Applied Microbiology International, the Embrace Center at the University of Washington, and Science in Seattle. And again, a big round of applause for our speakers today and our session chairs, who did just an incredible job. To name the folks involved in this behind the scenes here and also on camera who helped out throughout this course and symposium. We had Crystal Perez, Jacob Kevin, and Noah Rappaport, our session chairs today, as well as all of our TAs and instructors, Alyssa Easton, Alex Carr, Nick Quinn Bowman, and Carl Geiser. And of course, our administrative staff who helps make this all happen, our coordinator, Dominic Lewis, Allison Kudla, Joe Meister, Audrey Hubbard, Erica Krager, Ray Rosario, and Thea Swanson. We couldn't have pulled this off without all of the help of this really dedicated team of people. As a nonprofit, we rely on the generous support of donors. If you like what we do here in our educational programming and you got something to throw our way, there's a link at the bottom of our website if you want to donate to ISB. And you can visit ISB and learn more about us at isbscience.org. All right, that's all I got to say. We hope you enjoyed it. We hope you have a great rest of your day. Like I said before, in the last couple of days, we're probably going to wind down our course. This is probably the last year we'll do the course, at least for a while. We might reboot it in the future. We do plan, however, to continue to do these virtual symposia. So stay tuned for the 2025 ISB Virtual Microbiome Symposium, and have a great rest of your year. Thanks all.